Okay, so guys, our topic for this week is an overview of the methods used in histology. Okay, so what are the different methods, okay, that we will be discussing for this week? Okay, these include now microscopy. Okay, so we will have a discussion of microscopy. Other than that, we have the preparation of tissues. So here we will be talking about the different steps in preparing now a tissue slide. Okay, and then we also have autoradiography, cell and tissue culture, enzyme digestion, enzyme chemistry, and of course we have visualization of molecules using techniques like immunohistochemistry and hybridization techniques. Okay, so hold on, techniques. Okay, so these are the topics, okay, that we will be discussing, okay, in this chapter. Okay, so we have an overview of the methods used in histology. Okay, so to start with, okay, with our discussion, let's start or let's begin with microscopy. Okay. So guys, no, of course, the most important tool, okay, that we are using in studying tissues, okay, especially, for us medical laboratory technicians or technologies, okay, we have this tool, okay, that is really important. Okay, and of course, that is our microscope. Okay, so today we will be discussing about microscopy. Okay, the different types of microscope. We have your light microscope. So we will be enumerating the different types of light microscope, like your bright field microscope, your polarizing microscope, interference microscope, and other types, okay, of light microscopes, okay? And of course, we will be discussing about the electron microscopy. Under that, we have our scanning electron microscopy or your SEM and your TEM or your transmission electron microscopy, okay? So guys, no, when we say microscope, okay, I believe that okay, the definition of or you know what a microscope is. Okay, so it's an instrument that magnifies an image and of course allows visualization of greater detail that is possible or yeah, that is possible with the unaided eye. Okay, so it means that with the use of our microscope we can see even the smallest objects, okay, na kayang makita ng mata natin. Okay, so as small or smaller than dust particles, kaya natin silang makita, of course, with the aid of our microscope. So for microscopes, we have two general types. A microscope can either be simple or compound. Okay, so guys, now when we say simple, we're referring to something that is of single lens, and a very good example of this is yung magnifying glass natin. So your magnifying glass is an example of a simple microscope. And of course, compound microscope, okay, this talks about the microscopes, okay, containing systems of lens or lens system. Okay, so ibig sabihin multiple yung kanyang lens, and a very good example, of course, yung ginagamit natin sa ating laboratory. Okay, why? Bakit multiple lens? Other than the ocular, we have there the objective lens. Okay, so that's your compound microscope. Okay, so guys, no, I will be discussing with you mamaya, okay, yung different types of our light microscope. Okay, they are actually under your compound kasi they contain multiple lens. Okay, so of course, but before moving on to the next slide, okay, I have here the link, okay, that you can click for you to read more about the light microscope, its definition, the principle, types, and parts of the magnification. So you can click the link now, or you can visit, okay, the same link under the self-directed activities or suggested self-directed activities section. Okay? So in the online module. Okay, so this time let's move on to the next slide. So guys, now we have here the resolution, okay, of the eye and that of the instrument. Okay, so if you can see here, the human eye can have a resolvable points, okay, at about 0.2 micrometer. Ibig sabihin, guys, no, at um, 0.2 micrometer distance, kayang makita ng mata natin kay kung yung dalawang object ba na to ay separate objects or these are two objects. For example, these are two objects, okay, as small as 0.2, okay, micrometer, millimeter I should say, okay, kaya ng mata natin na makita kay yung dalawang object as separate objects, okay, two objects as separate objects, okay. But if you can see, when we're using yung ating mga microscopy techniques, okay, or yung mga microscopes natin, or yung mga instruments natin for microscopy, we can have, okay, the um, ability to see now smaller objects, minute objects. Okay, like for example, when we're using bright field microscope, so the distance between the resolvable points is at 0.2 micrometer. Eh, yung human eye natin, yung distance, okay, na nakikita natin to appreciate that these two objects are separate objects is at 0.2 millimeter. But with your bright field microscope, siguro kapag ka nakita mo yan, magkadikit, okay? Ang distance nila is at 0.2 micrometer. That's the resolvable point. With your human eye, hindi mo na ma-appreciate, okay, na dalawang objects sila. So, para magkakaroon ka ng impression na, okay, this is just an one object. Okay, but with the use of your microscope, makikita mo that they are actually separate objects. Okay, because you can see resolvable points at 0.2 micrometer. Okay, with the aid of your microscope. So with your electron microscopes under your SEM and your TEM, mas maliit pa na distance kaya niyang i-resolve. Ito in micrometer, this is at um, raised to negative 1,000. This one is raised to the negative 9,000. Okay, I'll call this one distance. So imaginein it's 2.5 nanometer. So if this is very, very minute object or these are very minute objects, kaya mo siyang makita, okay, as different objects kasi yung kanyang resolvable point, okay, even if the distance is at nanometer, you can see it, okay, or you can see these objects as separate objects because of your SEM and TEM's resolvable points, which is at 2.5 nanometer. So kahit in nanometers pa or 3 nanometers pa yung distance, okay, nung ating dalawang object na ito, makikita mo pa rin, ma-appreciate mo pa rin na they are two separate objects. Bakit? Kasi the microscope you are using is capable of um, visualizing now, okay, resolvable points as short as 2.5 nanometers. So when we say nanometer, that is about a 10 raised to 9, right? Negative 9, okay, na distance in meter, okay? Yeah, negative. Okay. So next, we also have the temp. For the theoretical, it's usually at 0.05 nanometer, but in tissue sections, okay, it actually resolve points at about 1.0 nanometers. So imagine, okay, how small that distance is. That is about 1 times 10 to the negative ninth of a meter. Okay. And then for atomic force microscopy, mas, okay, maliit na naman to, it is in picometer. Okay? So imagine at 50 picometer, ma-appreciate mo pa rin yung maliliit na organisms, okay, as two different microorganisms or separate microorganisms. Okay? Instead of just seeing them as one. Okay? Na-appreciate mo na, okay, they are different organisms or they are separate organisms or microorganisms because okay the microscope we are using is making you able now to see the objects at a resolvable points of 50.0 picometer. Okay, so yan. So this time, let's move on. Okay, so we have here no the properties, okay, of our light microscope. Okay, so we have what we call as the resolving power. So guys, no, when we say resolving power, we're talking about the smallest detail, okay, that a microscope can resolve. So yung pinakamaliit, okay, na detalye na kaya niyang i-resolve. Like for example, yung sabi ko nga kanina sa inyo, yung dalawang object na magkadikit. So, kaya niyang ipakita sa'yo na kahit maliit sila, okay, ma-appreciate mo that they are actually two objects, okay, that are, they are actually separate objects, okay? Instead of seeing them as one object lang, kasi sobrang liit nga nung distance, di ba, na kayang makita nung human eye natin. Okay, but with the use of your microscope, makikita mo na, okay, they are really separate objects. Okay, so that's the resolving power. It is, okay, the smallest, or it denotes, okay, that the smallest, okay, detail a microscope can resolve. Okay, so guys, no, there's actually these things, okay, that you have to remember. We have the difference between your resolution and your magnification. So, adding pag sinabi nating resolution, okay, we are talking about the ability of the microscope, hold on, microscope to distinguish detail. Okay, so we're talking about the ability of the microscope to detect, okay, two objects as separate objects. That's resolution. Okay, and when we say magnification, we are talking now about the ability of the microscope, hold on, microscope, to see small objects as, objects seem larger. Okay, so it's the ability of the microscope to see small objects seem larger. Okay, so these are the terminologies that you have to remember about your microscope. We have resolution. So when we say resolution, we're talking about the ability of the microscope to distinguish detail. Okay, is your microscope capable of letting you know, okay, that these objects, minute objects, are actually two separate objects. Okay, so that's the resolution. Okay. And when we say resolving power, we're actually talking about the smallest detail, okay, that this microscope can resolve. So it's kind of related to this resolution. Okay. And then we also have your magnification. When we say magnification, we're talking about the ability of the microscope to see small objects seem larger. Okay. So imaginein mo, like for example, using a magnifying glass, di ba? Yung isang, for example, isang seed siguro pag walang magnifying glass, ganito lang siya kaliit. Pero pag naglagay ka ng magnifying glass or tinutok mo siya sa magnifying glass, ganito na siya kalaki. So, magnify from the word magnify, okay? Lumalaki yung isang object. Okay? That's your magnification. And for your compound microscope, okay, that is true, of course, the aid of our objective lens. Okay? The ocular plus your objective lens. Okay? So the microscope used by most students and researchers is the bright field microscope. Okay, and it, of course, it has the following parts. We have the light source. From the term itself, light source, it's the one providing, okay, yung source ng ating light. We also have the condenser. Okay, so we can adjust no the intensity of the light coming into, okay, our object of interest, okay, or yung ating specimen. The stage that holds the microscope, the microscopic slide or the slide, okay, that you are viewing, the objective lens, okay, that further magnify, okay, yung image, okay, na nakikita natin dun sa ocular. So, di ba kung malala nyo, your objective lens has the following magnification. We have 4 times magnification. We have your 10 times magnification, your 40 times magnification, and then, of course, we have your 100 times magnification. Okay? So, your 4x is actually your scanning, and then we have your halo, objective, and then your high, and then of course your OIO or your oil immersion. So to get the total magnification, we just simply multiply these values, okay, with the value or the magnification times of your ocular or your eyepiece. So if you're viewing the object under your scanning, okay, objective, that's actually 4 times magnification. Just have to multiply this with the 10 times magnification of your eyepiece. So that will give you a total magnification of 40 times. Okay. So that's for your objective lenses and your ocular lenses. Okay? So for the OIO naman, the total lens magnification is at 100 times. And if you're going to view your object under your OIO, okay, the total magnification is actually 1000. So you just have to multiply this. Okay, the magnification of the lens times the magnification of the ocular, that will give you 1000. Okay? And then of course, your ocular lens, okay, kung saan po tayo sumisilip, where we peep, okay, where we observe yung ating specimen at view. Okay? So the magnification for our ocular lens is 10 times, okay, or 10x. Okay? So those are the basic parts, okay, of our microscope. So guys, no, I hope no you'll have a review of all of these parts, and try also not to get a copy of the picture or poster of a microscope and memorize the different parts as well as their functions. Okay? So, mamaya makakita natin yung parts ng microscopes natin, and I will let you do simulation online. So, e-learning natin, we will have a microscopy simulation. So, I will be or you will be seeing, okay, the link later, so you can access it while listening, okay, to this lecture, or if not, you can visit, okay, the microscopy simulation section in your online module. Hold on. Okay. So guys, we have here the parts, okay, of our microscope. So these are the basic parts. What I want you to do is, of course, to get a copy of this. Okay, you can actually get different no pictures coming from the internet. So please memorize the different parts, especially the most common or the basic parts, and of course their functions. Okay, and of course for you to have a practice, okay, or a simulation of microscopy technique, you can click the link, okay? Or kung ayaw nyo namang gawin na ngayon siya, you can actually visit the same link doon po sa inyong microscopy simulation section sa inyong online module. Okay? So next, this time, let's discuss no, the different types of optical systems. So we'll be talking about the light microscopes. Okay, so guys, no, first is we have, of course, the phase contrast microscope. One of the highlights, okay, for your phase contrast microscope is that, of course, we use them, okay, for unstained cells and tissues. Okay, so this type of microscope is very beneficial when we're examining or viewing cells that are living and are unstained. Okay, so we have two modifications of the phase contrast microscope. We have what we call as your interference microscope, or simply the phase contrast microscope, and the differential interference microscope. So guys, know your interference microscope or your phase contrast microscope allows quantification of tissue mass, while your differential interference mass microscope is useful for assessing surface properties of cells and other biological objects. So in the next slide, you will be seeing no pictures of specimens or cell viewed under your interference microscope and your differential interference microscope. So if you can see here, we have a picture showing you now a buccal mucosa cell. Okay, it's actually an epithelial cell coming from the human buccal mucosa, and it shows now the following structure. So it depends kung ano yung ginamit natin. So for under your DIC or your differential interference contrast microscope, or your DIC, ito siya guys. Okay, so 'di ba matandaan nyo kanina, sabi niya your DIC is very helpful, especially when viewing now or assessing surface properties. Okay, so this is actually the cell. So if you can see this cell here, okay, viewed under your DIC, it seems like it's something that is embossed. Okay? So makikita mo rin yung granularity dun sa surface ng cell, 'di ba? Parang magaspang yung dating niya. Okay? And of course, you can see yung how you call this one, yung bulk, okay, appearance or bulky appearance nung nucleus natin. So, may umbok siya. So, parang embossed yung dating niya. Okay? And of course, we have there yung mga dots or yung rough surface dito sa cytoplasm. Those are actually your cytoplasmic inclusions. And of course, presence of some bacteria in the upper surface or on the upper surface. So, guys, no, ito yung advantage nung ating differential interference contrast. Okay? As mentioned a while ago, okay? So it helps now in assessing the surface of the cell, okay? While this one naman is under your phase contrast or your interference microscope, okay? So kung makikita nyo, it shows an image, okay, that uh, has a pronounced halo. Kung mapapansin nyo, di ba, parang ito, may halo siya sa gilid, yung white corners niya. Okay, that's the halo. And then not only in the periphery of the cytoplasm, kahit doon sa periphery nung ating nucleus. Okay? So, kung makikita natin guys, no, these halos are actually not present doon sa inyong differential interference. Walang halo appearance. Pero na-appreciate natin yung umbok, 'di ba, nung nucleus mo, ay yung granularity, okay, nung surface natin na hindi natin na-appreciate dito sa ating phase contrast. So again, your phase contrast naman or your interference phase contrast microscope is used naman for the quantification, okay, of your tissue mass. Okay, so again, these are actually images, okay, viewed under phase contrast microscope. For letter A, it's under your differential interference. And for the second one, it's under your interference or simply phase contrast microscope. Okay, so this is actually an epithelial cell coming from the buccal cavity or buccal mucosa. So next, okay, we have another type of microscope. We have the dark field microscope. So guys, now we're just having an overview, okay, of the different methods in histology. We will not go deeper in our discussion because these are the things that you'll be discussing soon dun sa mga higher subjects ninyo. Okay? Para magkaroon tayo ng basic knowledge pagdating nyo sa mga major subjects. Okay, I know what this microscope's for. Okay? So for dark field microscope, it's usually useful for examining now autoradiographs. Okay? Other than that, it's used now in examining urine for crystals. So mamaya makikita nyo yung image, okay, ng crystals na na-view natin no doon sa ating urine. Those are actually cystine crystals viewed under your dark field. Ma-appreciate nyo sila. And of course, it's actually used now in the diagnosis of your syphilis. Okay, so nakikita natin yung microorganism that cause syphilis. That's actually your Treponema pallidum. Okay, or yung inyong syphilitic microorganisms. So we can appreciate the appearance as well as the movement, okay, in your dark field microscope. Okay, so your dark field microscope, guys, no, it's not only beneficial for your autoradiographs, okay, but again, it's beneficial as also in microbiology. Bakit? Kasi with your dark field microscope, na-appreciate natin yung shape niya. Kung rods ba, yung shape ng bacteria mo, kung curved rods ba sila, okay, and even their movements. Okay, so that's your dark field microscope. So bakit pala siya tinawag na dark field? Kasi dark yung kanyang paligid. So the object will appear bright, okay, against a dark background. The field or the background is dark. Kaya siya tinawag na dark field. Okay, kabaliktaran ng bright field microscope natin. Okay? So we have here a picture. Okay, these are actually pictures showing you now your Treponema pallidum viewed under your dark field microscope. And of course, we have here cystine crystals from the urine of a patient suffering from cystinuria. Okay, so kung makikita nyo siya, ma-appreciate nyo yung piatos-like appearance, okay, nung ating crystal. Okay, so this is under your dark field. So if you can see the microorganism or the crystals, okay, they appear bright, okay, against a dark background or a black background. Okay. So that's an example of your dark field microscopy application. Okay. So other than your dark field, we also have your fluorescence microscope. Okay. So guys, sa ulitin ko, all that we are discussing now are examples of your light microscope. So we have started with your phase contrast, and then we have your dark field. Now we have your fluorescence microscope. Okay. So guys, now your fluorescence microscope is very important, especially when displaying autofluorescent molecules. Okay? So these autofluorescent molecules, sometimes we call them yung pinaka-common na ginagamit even in your serology, we call these as your fluorochromes. They act as stains, okay, to view now antibody and antigen complexes or binding. Okay, so they are your fluorochromes. Mamaya, you will be introduced of the most commonly used fluorochromes, okay, in the laboratory and ano yung kulay nila, okay, when viewed under your fluorescence microscope. Okay, so your fluorescence microscope, again, is used to display autofluorescent molecules such as your vitamin A and some neurotransmitters. Okay, but of course, in this subject, in histology, or for example, in histopathology, it's very helpful in the detection of antigen. Okay, hold on. Antigen or antibodies. Okay, so we use the antigen or your antibodies. They can either be polyclonal or your monoclonal antibodies to detect now yung mga antigens natin, okay, dun sa tissue. Like for example, gusto nating malaman kung yung brain tissue na ito coming from a patient, okay, has this rabies antigen. So we're going to detect it using antibodies, okay, that are labeled with your fluorochromes. So mamaya ma-appreciate natin kung paano ba to ginagawa. Okay? So guys, no, we have here, okay, a slide showing to us, okay, a picture of your positive direct fluorescent assay and your negative direct fluorescent assay. So these pictures, okay, are actually pictures, okay, of what have been observed, okay, dun sa inyong um, fluorescent microscope. Okay, this was actually observed under your fluorescent microscope. Okay, so guys, no, this is the positive DIFA or your direct fluorescent assay, and this one is negative. So kung makikita nyo, walang bright objects, okay, dito sa negative DIFA natin. Okay, but here you can see apple green, okay, a fluorescence. Okay? So, bakit? So, ano ba to? Okay? So, guys, no, this is actually the direct fluorescence antibody test, okay, of brain tissue from animals. Okay, that is suspected of infection with your rabies. So, paano to? Okay? So, guys, no, since this is a direct technique, gumamit sila ng antibody, okay, to detect now the presence of these rabies antigen doon sa brain nung tao. So, paano yon? So, we have here a slide. Okay, fixing the slide is a brain tissue. Example lang to So, this is just for the sake of illustration. So guys, no, here in this tissue, we have this rabies antigen. Kunwari, ganyan yung shape niya. Okay? So this is the rabies antigen or these are the rabies antigens. Okay? Ganyan yung shape nila. Okay? And then, we use now, okay, antibody. Again, the name of our antigen is rabies antigen. Okay, right? Okay, that's the antigen. Okay, rabies antigen. Sila yan, yung mga triangle. Okay, and then we also have these antibodies. Yung reagent antibodies natin, gagamitin natin sila to detect now if there's really presence of your rabies antigen doon sa tissue. So guys, no, yung ating mga antibodies na to, we call them as your anti-rabies antibodies. Okay? Anti-rabies, sorry. Okay. Anti-rabies antibodies. Okay? So they are your anti-rabies antibodies. So guys, no, yung mga anti-rabies antibodies natin, they're actually against the rabies antigen, right? So your rabies antigen have okay, or rabies antigens have antibodies against them. And these are your anti-rabies antibodies. Okay? So, upon adding, okay, dun sa ating specimen, yung ating reagent antibodies, ibabind niya yan. Kasi kung ito yung rabies antigen natin, 'di ba? This antibody, sabi natin, is directed against the rabies antigen. So dahil may present na rabies antigen, magbabind ngayon sila. So we have now what we call as your antigen. This is your antigen and this is your antibody. Antigen, antibody, complex. Okay? So meron kasing binding nung rabies antigen natin plus the anti-rabies antibody natin. So we call this as your antigen antibody complex. Okay? So ganyan siya, guys. Okay? Yan. Sorry. So guys, no, in the fluorescent assay, they're actually using fluorochromes, okay, in labeling these antibodies para ma-visualize natin kung may binding na nangyari kasi nagkakaroon tayo ng binding, okay, kapag present yung antigen sa tissue. Okay? But if, for example, dito sa negative natin, okay, meron tayo ng tissue, wala nung rabies antigen, kahit mag-add ka ngayon nung antibody mo, okay, wala siyang pagbabindan. Okay, tama? So your antibodies will be unbound. Okay? And in your fluorescent assay, we have this how do you call this one? This is step where we do washing. The purpose of washing, okay, after incubating now the reagent antibodies with that of the tissue is, of course, to remove the unbound, okay, antibodies. Unbound, ibig sabihin, yung mga antibodies natin na hindi nakadikit sa antigen. Since dito sa tissue na to, wala naman tayong, how do you call this one, rabies antigen, so hindi magbabind ngayon or walang antigen na kung saan pwede mag-bind si antibody natin. So, they will remain unbound. So, if you wash them, matatanggal sila. Okay, matatanggal sila. And then, if you view them or if you view the slide doon sa microscope mo, negative, wala kang makikita. Ma'am, ano ba yung nakikita? It's actually the fluorescence, okay? Coming from the fluorochrome label na ginamit mo to label your antibody. So, anong ibig kong sabihin? So, guys, no, yung antibody, reagent antibody na ginagamit natin to detect these antigens, they're actually labeled. So, meron silang label. Okay? So, itong label na ito ay yung mga tinatawag nating fluorochromes. So, guys, no, what are the different, okay, fluorochromes that we're using in your fluorescent assay? So, I'll be mentioning, no, yung mga common lang. So, yung mga, sorry, yung mga common fluorochromes lang na ginagamit natin, guys. We have, for example, your acridine orange. So, the acridine orange, it gives a orange fluorescence, okay, under your fluorescence microscope. We have also your auramine O. Your auramine O, sorry, has a yellow color under your fluorescence microscope. Paki-correct. Hold on. Auramine O. Okay. Yellow fluorescent siya. We also have your rhodamine B. So, ang kulay ni rhodamine B, we have bright red. Okay. And then last, we have your hold on, fluorescein thiocyanate, okay? Or this is sometimes called as your fits, okay? So, isa to sa pinaka-common na ginagamit natin and it gives a color of apple green, okay? Fluorescence, okay? So, dito sa example natin, makikita nyo, it's actually apple green, right? So, ang ginamit dito na label is actually your fluorescein isothiocyanate, okay? So, guys, no, yung ginamit nila na label para dito sa mga antibodies natin, okay, ay actually fits. Okay? So, ito sila, kunwari yung mga bilog na yan. So, kapag nag-bind si antibody sa antigen, guys, sasama din pati yung label. Okay? Kasi labeled yung antibody natin. So, meron siya niyan. Okay? Anong kulay? Apple green. That's why, kapag, if for example, no, the antigen, ulitin natin tong, wait lang. Okay? So if for example, this is the tissue, okay? And the tissue actually is containing now the antigen. your antibody will bind this. Your labeled antibody. Bakit labeled? Because your antibody is actually labeled with your fits. Okay? So, ibabind niya. Itong antigen ngayon or yung antibody ngayon, reagent antibody, magbabind sila ngayon dun sa antigen na nasa ating tissue. Okay? So, ganyan siya. But, if you're going to wash after incubating now the reagent antibody dun sa tissue natin, ang next step kasi doon is washing. Guys, no, pag wash natin, they will remain dito sa ating specimen. Bakit? Kasi nakakabit na sila doon sa antigen natin. Okay? They are already bound. Okay? So, kaya meron tayong tinatawag na antigen, antibody, complex. Pero sabi ko nga kanina, pag negative yan, if this is the tissue, at gumamit ka ng reagent antibody mo, okay, na fluorescent isothianate labeled din, guys, no, pag nag-wash ka, mawawash out, mawawala yung antibodies mo. Mawawala sila kapag ka nag-wash ka na. Bakit? Kasi hindi sila nakabind doon sa antigen. Walang antigen kung saan sila dapat mag-bind para hindi sila ma-wash out. Kaya pag view mo under the microscope, negative siya. Wala kang makikitang fluorescence kasi na-wash out sila. Okay? But if the tissue is containing antigens, rabies antigens, again sabi natin the antibodies will bind these antigens, and if you're going to view it under your fluorescence microscope, makikita mo yung apple green for fluorescence. Bakit may apple green? Kasi you have to take note, 'di ba, na retain yung antibodies natin, nakabind na sila sa antigen, yung antibodies na yon ay fluorescein isothiocyanate labeled or FITS labeled. So, ang nakikita natin actually is yung label nung ating mga bound antibodies doon sa antigen. So, this will be read as positive direct fluorescein assay. Ibig sabihin, okay, this tissue is actually containing, the rabies antigen. Okay? So, that's the use or application of our fluorescence microscope. Okay? So, maaral nyo to lalo guys pagdating nyo no sa histopathology or histopathologic techniques and cytology as well as in your serology. Okay? So, you'll be discussing these techniques. So, yung diniscuss ko lang sa inyo guys is direct fluorescence assay. Bakit tinawag siyang direct? Kasi guys, no, we're just using one antibody to detect the antigen, one antibody. Mamaya kasi pagdating natin sa immunohistochemistry, you will see there, no, merong technique na gumagamit ng dalawang antibody for the detection. And we call that technique as indirect. Okay, but for this one, it's direct because we're just using one antibody type, to detect the antigen. And it's actually an antibody directed against the antigen. So dito sa example natin, since our antigen is rabies antigen, then the antibody that we use, in detecting now these rabies antigens are your anti-rabies antibodies, and they are labeled. Okay, so ang label nila ay yung inyong FITS or your fluorescein isothiocyanate. Okay, so that's for your fluorescence microscope and the application we have in your fluorescent assay. Okay, so guys, now next we have the confocal scanning microscope. So your confocal scanning microscope allows visualization of biological specimen in your 3D dimension or in 3D dimension. Okay, so intandaan ninyo, it provides now an image of a biological specimen in a 3D, configuration or dimension. Okay, so it combines components of your light optical microscope with the scanning system. And what's good, with your confocal scanning microscope is that you're able to dissect the specimen. Okay, so pwede nating i-dissect yung specimen optically. And while dissecting, we can see yung mga minute microorganisms as we dissect it. Okay, so that's the advantage of our confocal scanning microscope. Okay, other than your confocal, we have your ultraviolet microscope. Okay, ultraviolet microscope kasi yung source niya ng light is actually from your quartz lenses. Okay, so guys, no, the image produced by your ultraviolet microscope depends now on the absorption of your UV light by molecules, okay, in the specimen. And it actually give you a resolution as small as zero point 0.1 micrometer and resembles the working, okay, of your spectrophotometer. So other than that, your ultraviolet microscope is able of recording now the images photographically. Okay. So yeah. Yan, isang use niya, okay, is the quantification of your DNA and RNA, okay, in individual cells and detection of your nucleic acids as well as the purine and the pyrimidine bases of your nucleotides. Okay, so that's for ultraviolet microscope. Nice to know. Okay, so next we have the polarizing microscope. Okay, so your polarizing microscope is actually a simple modification of the light microscope. Okay? Guys, no, di ba yung light microscope ninyo, it has the following basic types sa lens system niya. Meron siya nung ocular. So this is the ocular, for example. Under this, we have the objective lens. Di ba? Ito yung mga objective lens niya. Okay? And then we also have here the stage and ito yung ating ano to. This is the specimen and this one is the light source. 'Di ba? Ito yung light source natin. So diagram lang siya, guys. Okay? So again, this is the ocular. These are the objective lens. This is the specimen because this is the stage. Okay? And of course, we have here the light source. Okay? So guys, no, sabi niya kanina, yung polarizing microscope natin is actually a modification of our light microscope. Okay? Paano siya na-modify? Okay? So your polarizing microscope, okay, or in your polarizing microscope, they added, okay, the following features, okay, or structures. So first is we have, okay, the polarizer. So guys, no, between the light source, so this is the, hold on, ayan, dito siya. Okay, filter. May filter siya dito between, sorry, between the light source and the specimen and another filter here between the ocular and the objective lens. Okay, so guys, no, sorry, we have here the polarizing filter. Okay, ito siya. The polarizing filter is actually placed between the light source and that of the specimen or nung stage natin. Okay? And another filter, okay, is added and that's what we call the analyzer. Okay? The analyzer naman is placed between your objective lens and that of the ocular. So ito siya. Okay? So yun yung modification nung ating polarizing microscope. It's actually same as that of your bright field, okay? It's just that, okay, na-modify siya by adding two filters, the polarizer and the analyzer. Okay? So, the principle is based on the fact, okay, that highly ordered molecules or array of molecules can rotate the angle of the plane of the polarized light. So guys, no, we have here no an example of a picture showing you now, okay, yung difference kapag yung crystal ay view natin using your bright field microscope, this one, and using your polarizing microscope. Okay? So guys, no, siguro naman na-appreciate nyo, okay, or nakikita ninyo yung difference nung dalawa. Okay? So, these are actually uric acid crystals, okay, coming from the urine, okay, of a patient, okay. So kung makikita nyo, under your bright field microscope adding, the crystals, okay, appear this simple, okay, 'di ba? So we have that hexagonal structure, some needle-like sheets, mga ganyan. Okay, so brown lang siya, doesn't have any color pa. Okay? But with your polarizing microscope, makikita mo, ma-appreciate mo yung crystalline characteristic, okay, nung ating uric acid crystals. So we have there various no blends of colors. Okay, kung makikita mo, yan. Okay, ma-appreciate natin siya. And the colors is of course now depending on the rotation of light dun sa areas ng crystals na yon. Okay? So this one naman is a picture, okay? um, taken under your polarizing microscope. Okay? So, mas na-appreciate natin yung details, okay, ng ating crystals compared when using, okay, yung ating bright field. Okay? So that's again, or these pictures again, okay, a view under your polarizing, and this one is under your bright field microscope. So we're not just using it for crystals, no, sa ating urine, but we are actually using din ito, guys, no, dun sa mga how do you call this one, crystals sa mga body fluids natin like synovial fluid, okay, and other body fluids. Okay, so yeah. Okay, this time let's move on to the electron microscopy. Okay, so for the electron microscope, we actually have two types. We have what we call as your transmission electron microscope and your scanning electron microscope. So guys, no, sa tingin niyo, what's the difference between the two? Okay, your TEM or your transmission electron microscope uses now an interaction of beam of electrons that, okay, passes through the image, okay, or through the specimen, okay? And while your SEM naman, okay, doesn't pass through your specimen, but it gives you, okay, a view or a scan, okay, of the surface of the specimen. From the term itself, scanning, okay, electrons. So it enables you to scan the surface of your specimen, or for example, a microorganism you're viewing under the microscope. Ma-appreciate mo ngayon yung kanyang structures outside, or yung kanyang appendages. Like for example, if it is flagella, okay, yung flagellated yung microorganism mo, mas ma-appreciate mo yung kanyang flagella, okay, how they're connected dun sa kanyang cell membrane, okay, using your SEM. Okay, but with your TEM, you're not just appreciating no yung surface. It's actually the inner, okay, portion of that specimen or of that microorganism. So sa TEM, it's enabling you naman to observe structures or structures found, okay, dun sa inner portion ng microorganisms mo. Like you can appreciate no yung kanyang mga organelles, like presence of the nucleus, okay, presence of the mitochondria, okay, nung cell natin. Okay? So that's the difference between your TEM and your SEM. Again, when we say transmission electron microscope, it gets through the specimen. Okay? While for SEM, it doesn't pass through the specimen, but it enables you to scan, okay, the surface of your specimen. So guys, now we have here a picture showing you now the difference, okay, of your SEM and TEM, okay, when viewing now specimens. So guys, no, we have here a picture of your Escherichia coli, okay? So in picture A, this is actually a picture um, from or under, okay, your transmission electron, and this one is under your scanning electron microscope. So guys, now under your TEM or your transmission electron microscope, we can see here the presence of the organelles of our E. coli. Okay, it's actually, sorry, it's actually your E. coli here. Okay, dito sa arrow natin. Okay, siya yung nakikita natin dito sa arrow. Okay, and this is actually a mast cell. Okay, sorry for that. Okay, so again, this is a mast cell. Okay, and using your TEM, we can see now the internalized, okay, Escherichia coli in the mast cell. So this one, yung nasa arrow, this is an E. coli, okay? So we can also see the nucleus, okay, of the cell. Ayan, this is transmission. Kumbaga, it's showing you yung mga structure sa loob. Okay, that's transmission electron. For scanning naman, okay, it enables you to scan the surface of the cell. So we have here the mast cell, okay, and if you can see, your mast cell is coated with your bacilli. So ito sila. These are actually E. coli microorganisms. So kung mapapansin nyo, nandyan sila, okay, balot nila yung ating mast cell. Okay, binabalot nila yung mast cell. So it's just the surface, okay, of the mast cell. Ito yung loob niya for your transmission. Dito sa scanning, yung surface niya. Okay? So again, that's your transmission electron microscope versus your scanning electron. Okay, your transmission enables you to see inside, okay, to appreciate the organelles, to appreciate if there's ingested or phagocytosed microorganism, while your scanning electron microscope enables you to scan the surface. So for example, this one, there are attached microorganisms in the surface of the mast cell. Okay, so na-appreciate mo or nakikita mo yung surface niya. Okay, so that's SEM versus TEM. Okay, so guys, no, before moving on to the next slide, you may click the link now to be redirected in our video A. So your video A will present to you the different types of microscopes and how to use your light microscope. Okay, so just click the link for you to be redirected. If you do not want to watch yung video, okay, this time, and you want to watch it later, you can just visit the suggested self-directed activities section. Okay, same videos are there, okay, or the links of the videos are actually there, okay. So guys, no, let's have a review before we move on dun sa preparation of tissues, okay. So we have discussed kanina, no, yung different types of microscopes, okay. Generally, we have the light and the electron microscope. So under your light microscope, we have discussed there the different types. We have your phase contrast microscope. So under this, we have your differential interference and your phase contrast or your interference microscope. We also have your dark field microscope showing you now the pictures of our Treponema pallidum and cystine crystals, okay, against a dark background. And this is your dark field, okay? And then we also have your fluorescence microscope. So one of the most common use, okay, that we can encounter later sa higher subjects nyo is of course its use in detecting now presence of antigen or antibodies in the specimen. So we have here an example. This is the direct fluorescent assay of a tissue infected with rabies. Okay, so this one is the positive result. If you can see there, we can observe presence of apple green fluorescence. Why? Because the rabies antibody, antigen is present in this tissue. Okay, so the rabies antigen, um, we're actually uh, finding now the labeled antibodies. So the labels of the antibodies here, okay, is actually your fluorescein isothiocyanate. So it gives now a apple green fluorescence. So positive. Dito, negative tayo. Okay? And then other optical systems such as your confocal scanning microscope that provides us the 3D dimension image, okay, of our specimen. Okay? And it's also capable now of dissecting the specimen optically. Okay, and then we also have the ultraviolet microscope and then the polarizing microscope. So under your polarizing, we can observe your crystals. Okay, so this is a picture of your uric acid crystals under your polarizing microscope. Okay, and then, so again, those are under your light microscope. Okay, and then under your electron microscopy, we have two. We have your TEM and your SEM. So we have here the picture showing you now the difference, okay, between your TEM and your SEM. So under your TEM, we can see through, okay, the specimen. Okay, we can see if there are inclusions, if there are engulfed or phagocytosed microorganisms. Okay, under your scanning or your SEM, we can appreciate the surface of the microorganism or the cell, okay, that we are actually viewing. Okay, so that's your transmission and your scanning electron microscope. Okay, so guys, no, this time, let's move on dun sa ating preparation of tissues. Okay, so kung mapapansin niyo siguro before you visited the laboratory at nakakita kayo no nung mga tissue slides natin, 'di ba? We have this glass slide here, tapos meron tayong mounted tissue. Okay, for example, epithelial tissue. So guys, no, here in this next topic, we will be discussing how these, okay, tissue slides are produced. Ano yung mga steps na gagawin natin for us to produce this tissue slide, okay, for histology. Okay, so we are now discussing preparation of tissues. Hold on. Okay. So for the preparation of tissues, we have the following steps. These are actually the 10 basic steps, okay, that we perform in preparing tissue slides. And guys, no, you will have a thorough discussion of these pagdating ninyo sa histopathologic techniques and cytology. Dito, overview lang, okay, yung gagawin natin. We will not discuss everything, okay? Kasi sa histopath ninyo, per chapter ito eh. Dito, we will just define the step and try to determine lang yung most commonly used agents. Okay? So guys, no, the first step is of course fixation. Okay? So the um, fixation process, okay, the purpose of that is of course to preserve, okay, yung ating specimen. So, our specimen will be soaked or immersed in a solution and this solution is what we call your fixative. Okay? So, sa laboratory, ang pinaka-common na ginagamit natin ay yung mga chemical fixatives natin. Okay? And the best general, okay, ulitin ko, best general fixative is actually your 10% hold on, neutral buffered formalin. Okay? Guys, tatandaan nyo, best general fixative is your 10% neutral buffered formalin. Okay? Siya yung ating madalas ginagamit, okay, sa laboratory. It's usually at the 10% concentration. Okay? Okay? So, siya yung pinaka uh, common na ginagamit natin. It's actually the best general fixative. Okay? So what's the purpose, okay, of fixation? Of course, the purpose of fixation is to preserve the cell and tissue structure. So isipin mo, how will you examine now, okay, a tissue or for example in histopath, how will the pathologist do proper diagnosis if naagnas naman na pala yung tissue na ginagamit mo? So, marami nang nangyari, okay, dun sa tissue that's not actually related dun sa possible disease, okay, na gustong i-diagnose ng ating patho. So, we have to preserve it, okay? That is okay, preserving the tissue in a lifelike manner. Kung paano yung tissue natin nung nasa katawan pa siya ng buhay na tao or yan, kung nung nasa katawan pa natin siya or nakakabit siya, preserve natin yung itsura niya as well as the processes. Okay, ina-arrest natin. Okay? So again, the purpose is to preserve the tissue by okay, avoiding now tissue digestion, okay, that are actually released kapag ka namatay yung tissue, okay, there will be release kasi ng mga enzymes dun sa mismong cells natin. And these cells will cause na what we call as your self-digestion or autolysis that will lead yung tinatawag nating agnas. Okay? So, pine-prevent natin yon. That's to preserve, okay, the cell. Again, the purpose is to preserve the cell in a lifelike manner. Okay? So next, and after fixation, guys, we have what we call as your dehydration. So guys, no, from the term itself, when we say dehydration, we're talking about the process where we remove, okay, the intracellular and the extracellular water in the tissue. Okay? What's the purpose of that? The purpose of that is to prepare the tissue, okay, to absorb or for the tissue to be ready, okay, in absorbing now the different solutions that we will be using in the subsequent processes. Okay, so in the dehydration process, how we're going to do that? Okay, we do that by transferring the tissue in a series of increasingly concentrated alcohol solution. So guys, no, in the dehydration process, okay, yung pinaka-common na series na ginagamit natin is that we usually start at 70%, then we have 90 or 95%, and then we have 100% or yung tinatawag nating absolute alcohol. 100% or absolute alcohol. So 70% alcohol. Then after that, after a few minutes or hours, we transfer it to 95% and then we transfer it to absolute alcohol or 100% alcohol. So usually 2 to 3 changes ng 100% alcohol. Okay? So ganun natin siya ginagawa. And again, the purpose, okay, of transferring that in or yeah, the purpose of immersing your tissue in The alcohol solution is to remove, okay, the intracellular and the extracellular water. Pag tinanong mo, ma'am, bakit increasing concentration? Hindi ba pwede, ma'am, na diretso na lang tayo sa absolute alcohol? Hindi. Kasi if you'll do that, you will increase the chance of tissue distortion. So we start sa lower concentration muna, then gradually papataas hanggang makarating tayo sa absolute alcohol. Okay? Or yung 100% concentration natin. For small specimen, they start no sa lower concentration usually at 30%. Okay? So depende sa size yan. Okay? For medium size or for general procedures, they usually start at 70. Pagka sobrang liliit, they lower, okay, yung concentration kung saan sila mag-start. Usually at 30%. Okay? 30, 70, 95, tapos absolute. Okay? So guys, now what's the commonly used alcohol for dehydration? It's actually your ethanol. Okay? Ethanol. Okay? So that's for your dehydration. Again, what's the purpose of dehydration? To remove the intracellular and the intercellular water from the tissue to prepare it, okay, in the subsequent processes, or to prepare it, okay, in absorbing, okay, the different solutions will be using, okay, dun sa next processes natin. Okay? So guys, so next procedure is your clearing. Okay? Guys, no, yung clearing natin, this is also known as your dealcoholization. Dealcoholization. Okay, so what do you think is the purpose of your dealcoholization or your clearing? From the term dealcoholization, the purpose is, of course, to remove the alcohol. Okay, again, the clearing process is used to remove the alcohol, okay, from the tissue. Saan galing yung alcohol? From our previous step, which is yung ating dehydration. 'Di ba sa dehydration ang ginamit natin alcohol. So to remove the alcohol, we use now yung tinatawag naman nating clearing agent. So guys, no, the most commonly used clearing agent is actually your xylene. Okay, your xylene. Okay? So that's for the clearing. Okay? Guys, no, ma'am, bakit the purpose of this process is to remove the alcohol? So we call it dealcoholization. Bakit clearing? Bakit siya tinawag na clearing? Mas akma naman yung term na dealcoholization. Because, guys, no, most of our clearing agents, if not all, okay, literally makes the tissue clear. Okay, they have high refractive indices. So yung mga ginagamit natin na mga clearing agents, if not all of them, most of them, okay, literally make the tissue clear. Okay, kaya tinawag din siyang clearing. Okay? Ibig sabihin, pag kiniklear niya yung tissue natin, mas madali siyang i-visualize later doon sa microscope natin. Okay? But again, it's more known kasi, okay, itong step na to or itong procedure na to, mas kilala sa term na clearing. Okay? But again, it's actually your dealcoholization. Again, the purpose is to remove, okay, the alcohol from the tissue. And saan galing yung alcohol natin? Galing siya dun sa dehydration process natin. Okay? So the most commonly used clearing agent, it's your xylene. Okay? So next, we have your infiltration. Guys, no, your infiltration is also known as your impregnation. Why? Because in the infiltration process, what we're going to do is to infiltrate the tissue with a substance or with paraffin. Ang most commonly used, okay, the most commonly used medium for impregnation is your paraffin. Okay, paraffin wax. Okay? So guys, no, ano ba yung purpose ng ating infiltration or yung ating impregnation? Okay? So guys, in the infiltration or in the impregnation method, okay, or process, we're actually, okay, filling yung spaces dun sa ating tissue. So if for example, this is the tissue, di ba tinanggal natin yung mga intracellular and intercellular water. Okay, so tendency, between cells, merong mga spaces yan. Okay? Ba may mga spaces, okay? And if that spaces, okay, won't be filled, ang tendency magiging crumbly yung tissue natin. So what we're going to do is to infiltrate that with your paraffin wax to make the tissue firm. Again, the purpose of your infiltration and impregnation is to make the tissue firm. Okay? That is to infiltrate the spaces with your paraffin wax. Okay, so the most commonly used medium for infiltration or impregnation is your paraffin wax. Okay, so guys, no, um, yung ating paraffin infiltration or impregnation, we usually use naman 2 to 3 changes, okay, of paraffin wax. So, nilulublob natin or binababad natin yung ating tissue for hours, okay, doon sa ating beaker or container that contains melted paraffin. Okay? So, ma'am, ano bang purpose, okay, nung, or what do you think is the principle behind the infiltration and impregnation? Something like this, okay? So guys, no, have you tried cutting, okay, a pork, yung karne ng baboy na hi- frozen at yung karne ng baboy na hindi frozen? Anong karne or alin sa kanilang dalawa yung madaling makapag-produce ng cubes? 'Di ba mas madaling hiwain pag frozen? 'Di ba, yung frozen kasi very firm siya. Unlike 'pag hindi siya frozen, sa Ilocano loppoy, yung parang, crumbly siya, 'di ba? 'Pag ginalaw mo mataba siya, 'di ba, shaky-shaky siya, okay? But with your frozen na pork, 'pag ginalaw mo siya, firm, very firm kasi siksik yung loob dahil nga puno nung ice crystals, okay? So ganun din dito sa infiltration. So imagine, 'pag kinat mo, 'di ba, yung frozen na pork, madali, okay, yung mag-cut at madali para sa'yo na makapag-produce ng cubes or or karne na or cuts na ang shape ay cubes, 'di ba? Unlike pagka hindi siya frozen, ang hirap mag-cut kasi magalaw yung karne. Okay? Ganon din yung sa infiltration and impregnation natin. That's to make the tissue firm to facilitate the cutting later of our tissue. Okay? So ano ba yung cutting na sinasabi ko? Mamaya, tignan natin pagdating natin dun sa process na yon. Okay? So guys, no, the next process, okay, or procedure after your infiltration or impregnation is what we call as your embedding. Okay? So kanina, we impregnated the tissue or we infiltrated it with your paraffin. This time, we're going, okay, to put our paraffin infiltrated tissue in a mold. Ano yung mold? 'Di ba? Para siyang, alam mo yung ice cubes nyo, 'di ba, when you're going to create or make some ice cubes, 'di ba, you're using a mold. Tapos lalagyan mo yung mold, 'di ba, ng tubig, tsaka mo lalagay sa freezer. So kung ang shape ng mold mo, for example, ay cube or square, magiging square din, 'di ba, yung inyong ice. Okay? So, meron ka ng ice cubes. Okay? Ganon din yung sa embedding. Okay? So, yung sa embedding, this is, for example, the mold. Okay? So, meron siya nung depression dyan kasi dyan malalagay yung tissue. So, if for example, ito na yung infiltrated tissue mo. Okay? Ilalagay mo siya dyan sa butas. Then, you will fill this, okay, with, again, melted paraffin. So, guys, no, usually, okay, the type of medium that we use sa impregnation, okay, is the same type we'll be using sa embedding. So, sa impregnation technique natin, gumamit tayo ng paraffin. In the embedding process, we're going to use ulit paraffin. So, ayan, if-fill natin ng paraffin to, yung paraffin wax, guys, kamukha nung kandila din ninyo yan. Okay? So, ganun siya. Pagka na-melt, di ba, ano siya, liquid solution. Tapos pag tumigas na siya, magiging solid siya. Okay, ganun din yung sa paraffin nyo. So, we are going to pour now melted paraffin wax dito sa mold natin. So, ilulubog natin si tissue. And then, we're going to let that cool. Okay? So, titigas na ngayon yung mold. Magkakaroon ka din ngayon ng square na. 'Di ba? Kung square yung shape ng mold mo, edi ganun din shape ng tinatawag mong tissue block ang tawag doon. So, kunwari, andyan na yung tissue mo. O, tumigas na yung mold. So, tanggalin natin siya from this one. This is the mold. So, tanggalin natin. Ito na siya. Okay? So, square na. Ang tawag dito, guys, is yung inyong hold on. Ang tawag dyan ay tissue block. Okay? So, yung tissue block ninyo, it actually contains now your tissue, paraffin-infiltrated tissue, and of course, the paraffin block. Ayan, paraffin yung paligid niyan, di ba? So, hold on. So, yung paligid nito, paraffin. Ito yung tissue mo. Oh. Okay? So, that's the tissue block. Again, guys, no, the medium that we use in the infiltration is the same medium that we use for the embedding. Okay? So, that's your embedding. Your embedding, guys, no, is also known as your casting. Okay? Casting. Okay? So that's your embedding. So guys, mamaya you will see another link. Okay, this is actually a process. Okay, of the tissue slide preparation. So you can watch it, you can click it, or if not, you can visit your suggested self-directed activity section. Okay, so next. Uh, Yan, trimming. Okay? So guys, no, minsan yung nagagawa natin na mold or tissue block, I should say, di ba ito na yung tissue block natin, nandun na yung tissue. Minsan, yung nagagawa natin guys na tissue block natin, hindi pantay, hindi siya perfect square. So that's the purpose of trimming. Okay? That is to trim, okay, the sides or the edges, okay, to create now, okay, a perfect block. Sorry, hindi pa rin siya perfect. So, dapat kwan siya, okay? So, in the trimming process, we're actually making, okay, the tissue block perfect, okay? So, in shape so that it will perfectly fit doon sa ating microtome. So, yung microtome, guys, no, yun yung ginagamit natin in cutting our tissue section. Okay? So, di ba may tissue block na kayo niyan? You will actually cut this, okay, to produce thinner section. And we use now yung microtome natin. Okay? We're actually using then yung microtome in trimming the tissue block. And the purpose of that, okay, yung trimming, other than, how do you call this one? Um, Trimming the edges is of course to expose the tissue. Kasi during the embedding process, natatakpan yan. Hindi ka agad litaw yung tissue mo. Usually natakpan din yan nung nasa gitna siya. So minsan nakatakip sa kanya ay makapal pa na paraffin. So for you to get a sample, you have to expose, okay, yung inyong tissue. And that's through the process of trimming. Okay? So, yan yung ating trimming process. Again, what we use is your microtome. Yung microtome natin, it actually contains now a knife. Okay, so yun yung pang-trim. So, if you have already visited the laboratory, meron tayong microtome nun. That's the rotary microtome. So, the most commonly used type is your rotary microtome. Okay, so your rotary microtome was actually invented by Minot. Okay, or rotary microtome. Okay, so next, we also have here the section cutting. So ganun din, in the section cutting, we're actually using pa rin yung ating microtome kasi siya yung pangkat natin. Okay? And the purpose of that is to cut the tissue block into thin films. Okay? Paano yun? Okay? So mamaya mapapanood nyo siya dun sa video para mas maintindihan ninyo. Pero bibigyan ko kayo ng example. Have you tried, okay, creating thin films, okay, doon sa inyong crayons? Or, for example, you have a crayon or kandila. Na-try nyo ba siyang kuskusin ng knife? Di ba? Pag kinuskus mo siya yan, magpo-produce siya ng very thin film. Okay, so parang ganun yung purpose natin dito sa section cutting. Okay, so guys, no, if itong tissue block na to, kunwari, ikakabit na natin siya doon sa tissue holder ng microtome. Kunwari, ito yung microtome natin. Dito sa baba niyan ay yung knife. Okay? Pagka inikot mo yung wheel, okay, tataas bababa itong tissue holder. So, pati yung tissue natin. So, ibig sabihin, lalapit at lalapit siya dito sa knife natin. Okay? So, anong product nun? Kasi nakakaskas yung iyong tissue block. Ang product nun, magpo-produce siya ngayon ng tissue ribbon. These are actually thin films. Okay? The tissue ribbons are thin films that, okay, have uh, impression, okay, of the tissue. Okay? So these are thin films, and if you observe, meron siya, or the thin films, no, have impression of the tissue. Thin impressions, okay, of the tissue. So meron siya niyan. Okay? So Kasi masyadong maliit ha yung cut natin. Yung cut kasi natin guys usually in micrometer, 10 micrometers. So manipis na manipis yan. Okay? So imaginein nyo to. Okay? So we have these pare-pareho kasi nga maninipis na impression, okay, nung inyong tissue. Okay, so this is your tissue ribbon. Okay, so guys, no, ngayon, ang gagawin mo dyan, if fish mo to, lalagay natin siya sa water bath. Okay, the water bath contains now a water, okay, that has a temperature that's about 6 to 10 degrees Celsius below, okay, the melting point of your paraffin. So, anong purpose ng pag-place natin ng tissue ribbon dito? So, kunwari, andyan na yung tissue ribbon natin. Lukot-lukot kasi yan, kulot-kulot, or fold-fold siya yan. So, the purpose of these water bath is to straighten or to stretch our tissue ribbon. So, pag nag-stretch na siya, you just have to get, okay, yung iyong slide and scoop mo siya. So, kunwari, ito na yung slide mo and you decided to get at least 3 impression of the tissue. Kunwari, ito yung tissue ribbon mo. So, in-scoop mo siya coming from the how do you call this one? The water bath. So, ito siya. Kunwari, ito siya. O, 3 yung kinuha mo na impression ng tissue. Ayan. Okay? And then, you're going to place this sa oven. The purpose of the oven, okay, is to melt now these excess paraffin. Ito, yung mga excess dyan. Para ang maiiwan lang, guys, is itong portion na to, kung saan nandun yung tissue natin. Okay? And also, to prepare, okay, our tissue for staining. Okay? So, ito na yung final product natin. Okay, andyan na siya sa slide. So, for it to be stained, you have to melt the excess paraffin wax. Kasi yung paraffin wax natin, it's immiscible with your stain. So, hindi mo ma-stain yung tissue kung hindi mo i-melt, okay, yung paraffin wax. Okay, you have to take note, in-infiltrate mo si tissue. Other than that, you've embedded it with paraffin. Okay, so you have to remove, okay, the paraffin. That's by placing it in the oven. Okay, so after section cutting, you're now ready to stain, okay, your tissue. Guys, no, in the routine histo techniques, okay, the stain that we usually use is actually your hematoxylin, and eosin. Okay, this is, again, the routine stain for histology. Not only for histology, even for histopathology, okay? It's actually regressive. Bakit siya regressive? Kasi it actually has this step where we employ alcohol. Okay? The purpose of that is to decolorize and to produce now contrast. Kasi dalawa yung ginagamit natin. Your hematoxylin, okay, stains now yung ating nucleus, and the eosin stains the cytoplasm. So guys, no, in your H&E technique, or staining technique, ano yung result natin? Okay? Guys, no, yung ating nucleus, it's usually dark blue or blue-black. Okay? Yung kanyang kulay. Sorry. Dark blue or blue-black. And the cytoplasm is pale pink. Okay? So again, the most commonly used staining technique, okay, in histology or in histopathology is your hematoxylin and eosin, or H&E staining technique. It's the routine staining technique, okay? And your hematoxylin, it's used for the staining of your nucleus, and your eosin for your cytoplasm. So the resultant colors, we have nucleus stained dark blue or blue black, and the cytoplasm pale pink. Okay, it's pale pink. So guys, no, your hematoxylin is an example of a nuclear stain. Of a natural stain. It's actually, okay, derived from a tree. It's a Mexican heartwood. We call this your hematoxylin campecianum. Hematoxylin campe. Yan. Okay, that's the hematoxylin. Sa net, minsan nakakakita kayo ng hematoxylin, H-A-E. Okay, so depende dun sa author. Okay, hematoxylin campecianum. This is the name of the tree, okay, where hematoxylin is derived. So it's an example of a natural dye. Okay, so guys, no, The dyes can be classified as either, okay, how do you call this one? Basic dyes or acidic dyes. Okay? When we say basic dyes, these dyes stain now basophilic structures of our tissue. Okay? While for your acidic dyes, they stain acidophilic structures. Okay? So guys, no, what are examples, okay, of basic dyes? Example of basic dye, dito nga is yung inyong hematoxylin. So sabi niya, a basic dye stains now the negative charge, okay, component, cell component. Your nucleus is negatively charged. Okay, so it's stained by a basic dye, and in your H&E technique, that's your hematoxylin. Okay, so guys, no, for your acidophilic components, okay, or yung mga cationic components natin, example, yung ating cytoplasm, they're actually stained by your acidic dyes, and an example of an acidic dye is your eosin. Okay? So, again, we have two types of dye according to the cell component they're staining. We have the acidic dye. Acidic dyes stain cationic components, okay, or acidophilic components, okay? And your basic dyes stains now the anionic components, okay? Your anionic components have affinity to basic dyes, thus they are called basophilic, okay? And an example is your hematoxylin. Okay, so that's staining. Again, ulitin ko guys, no, for the routine histo technique or routine histology or histopathology, the routinely used staining technique is your hematoxylin and eosin staining. Okay, so next, we have mounting. Okay, so guys, no, ito na yung slide nyo. Okay, yan. Na-stain na rin siya, may kulay na rin siya. Okay, ito na yung tissue. Kunwari may kulay na rin siya. Okay, so, pasensya nyo na yung drawing ko. Okay? So, may kulay na siya. So, okay lang din naman na i-view mo siya ng ganyan, pero hindi siya kasing linaw kapag ka meron ka nung cover slip. One of the purpose din kasi of the cover slip, okay, attached to it with the mounting medium is that it will have, okay, higher refractive index. So, mas maganda or mas clear siya tignan. Okay? Other than that, okay, kapag hindi mo tinakpan with a cover slip itong tissue mo, ang mangyayari, mag-fade yung kanyang stain. Okay? At pag in-store natin siya, for example, naka-expose siya, iniwan natin siya dyan, may tendency din na kainin siya ng mga daga. O kaya yung sa Ilocano quote, bau. Okay? O balakak sa ibanag. Okay? So, to prevent no fading of the stain and to protect our tissue, okay, dun sa slide natin, on the slide, you have to cover it using a cover slip. And of course, we can add, how you call this one? We can attach, okay, our cover slip using a mounting medium. So guys, now what's the purpose of mounting? Okay, so again, the one purpose is to increase, okay, the refractive index. Other than that, we also protect, okay, yung ating tissue, okay, from its stain, okay, na mag-fade. Para hindi mag-fade yung stain niya, we cover it, okay, with a cover slip. Okay, so guys, no, yung ating mga cover slip, guys, no, meron silang refractive index, okay, or their refractive index is at 1.518. Okay? At yung ginagamit nating mounting medium, ano yung purpose ng mounting medium? Kumbaga siya yung parang mag-glue nung cover slip mo dito sa slide mo para hindi siya natatanggal. Nakadikit na siya. Okay? Guys, no, yung mounting medium natin, kamukha siya ng parang, how do you call this one? Yung mighty band. Pero, of course, hindi siya mighty band, hindi mounting medium yung mighty band nyo. Okay? Parang ganun lang siya. Okay? So, yung mounting medium, di ba yung mighty band ninyo, pag tumigas siya, nagiging opaque yung kulay niya. Yung mounting medium, hindi. Pag tumigas siya, guys, clear pa rin siya. Parang kamukha nung ano ba, nail polish or yung cutex na sinasabi nila, okay? 'Di ba pag tumigas yung clear color, clear pa rin siya. Okay? Ganun din yung mountant natin or yung mounting medium natin. Okay? Guys, no, your sa histopath, ang minememorize natin usually sa mounting medium natin is yung kanilang refractive indices. Okay? The refractive indices of our mounting medium, okay? Or our mounting media are usually near, okay, that of our cover slip. So ang refractive index ng ating cover slip is 1.518. So our mounting medium, they are also at the range of 1.5 to something, okay? So yun yung kanyang refractive index, okay? So guys, no, the most commonly used no mounting medium, okay, we have your Canada balsam. This is actually derived from a tree known as your sorry, Albus balsamea. Okay? So guys, no, it's an example balsamea. Okay? So this is an example of a resinous mounting medium. Okay? So mounting media. Okay? Okay. So guys, no, yung Canada balsam natin, it's derived from a tree. So ibig sabihin, it's naturally sourced. Okay. So it's an example of a natural mounting medium. Okay. So the refractive index of our Canada balsam is usually at 1.54 to 1, sorry, to 1.55. So near the refractive index of our cover slip. Okay, it's a resinous mounting medium. So para siyang resin, malagkit siya. Okay, so that's mounting media. Okay, what's again the purpose of mounting? Again, to prevent the stain from fading or the stained tissue from fading its color, and of course to protect our tissue from scratches, okay, or to protect it and for it not to be eaten by your baug or yung inyong balakak or daga. Okay? So, yan. And then finally, of course, we have your labeling. Okay? It's a no-no, okay, for you not to label your specimen. Okay? Okay lang siguro, no? Yung, kasi even, di ba, sa lahat ng bagay, there should be label. Okay? Charot. I I believe yung iba sa inyo nakaka-relate, 'di ba? So yeah, pero joke lang, okay? Again, the last step is labeling, okay? So guys, no, if this is your tissue slide, we have here the tissue. We usually use tissue slide, okay, that has a frosted end, okay? So we just simply label this, okay? During the process, pag nag-staining ka pa lang, nagki-clearing ka pa lang, please label your slide using a lead pencil. Do not use, okay, yung inyong felt tip pen, okay, or marker. Kasi 'di ba in the process kasi gumagamit kayo ng xylene, alcohol, mabubura 'yon. So nagulat ka lang wala nang label yung specimen mo. So napagalo-halo mo na, hindi mo na alam kung kanino yung para kay kanino, 'di ba? So you have to use pencil. Pero pag natapos na yung staining mo at babasahin na lang, you can use a marker. Okay? That's to permanently label yung iyong slide. Okay? So, labeling. So, guys, no, in the labeling of specimens or tissue slide, for example, histology kasi ito, so you just, for example, if this is a tissue coming from the buccal cavity, for example. So you can just say epithelium, okay, buccal mucosa. Okay, so ano yung specimen mo na yan? That's for histology. But for histopath later, iba yun. Sa ospital, iba yung labeling na ginagawa nila. They label it using yung tinatawag nating accession number. Okay, yung number na naka-assign dun sa ating patient. Okay, dun sa ating registry. Okay? So, yun yung label nila for histopathology. But for histology, we just simply put kung anong type ng specimen ito. Is it simple columnar epithelium? Coming from where? Okay, the digestive tract, for example. Okay, so that's labeling. You have to label your specimen. Okay, hold on. Yes. Okay. So guys, no, we have here a picture showing you now a summary or a simpler diagram, okay, of our processes. Number one is of course yung letter A, guys. Anong ginawa niya? We have there a scalpel and the tissue. This is actually known as your grossing. Okay? It's actually the pathologist, okay? Siya yung gumagawa nito. He will pick no kung saang part ng tissue yung isa-sample nung organ yung isa-sample. Hindi naman yung buo. Okay? Kukuhanan niya lang yung area na suspicious. Okay? For example, of cancer. If for example, the patient is suspected of having a cancer, parang ganun. Okay? Then after that, they're going to fix this. Okay? So, yan, naka-immerse yung ating tissue sa solution. Guys, no, ano usually or the most commonly used fixative is actually your 10% neutral buffered formalin. Ano yung ratio? Guys, no, 20:1. Anong ibig sabihin ng 20:1? Ibig sabihin, this is your fluid to tissue ratio. Ibig sabihin, guys, yung volume daw ng ating fixative, it should be 20 times the volume of our tissue. Again, that's the fluid tissue ratio. It's 20:1. Okay? So, dapat ang volume ng ating ng ating fixative is 20 times than that of the volume of our tissue. And then after that, we have dehydration. So guys, now if you can see here, we have three containers. Bakit? Di ba, malala nyo, sa dehydration, using your alcohol or for example, your ethanol, specifically your ethanol, di ba we're using there increasing grade of alcohol. We do not, okay, immediately proceed with your absolute. So mag-start tayo, for example, 70%, and then we have 95%, then finally, we have your 100%. Usually, 2 to 3 changes din tong 100% natin. Depende dun sa protocol na ginagamit natin. Okay? You will know more of that pagdating nyo sa histopath. Okay? So, 3 changes. For example, in 70% solution, 1 hour siya dyan or 30 minutes. After that, ililipat niya siya dito. Tapos, ililipat niya ulit siya dun sa, ililipat niya from 70, ililipat niya sa 95. Then, after a few hours, okay, depende dun sa schedule na ginagamit, dun na sa absolute alcohol. Then, after that, you will put it in the clearing agent. Again, what's the most commonly used clearing agent, guys? It's actually your silane. Okay, your silane. What's the purpose of your clearing again? That is to remove now the alcohol, okay, that you use in the dehydration process. Okay, your clearing is also known as your dealcoholization. Guys, no, ano naman daw yung ratio? How or what is the volume, okay, of the solution used in dehydration and clearing? Okay, the ratio I should say, at 10:1. Okay? Both for dehydration and clearing. Anong ibig sabihin? The solution or the volume of your dehydrating fluid should be 10 times the volume of your tissue. Ganon din sa clearing. Okay? The volume of your silane should be 10 times the volume of your tissue. Okay? Then after clearing, the next process is your infiltration. Dadalhin mo na yung tissue dito. Okay? What's the purpose of your infiltration? This is also known as your impregnation. Okay? The most commonly used medium is your paraffin wax. Okay? So what's the purpose again of your infiltration or impregnation? That is to make your tissue firm. Okay, that's to facilitate cutting of our tissue later. Okay, and then after your paraffin infiltration or impregnation, the process or the next process, okay, would be embedding. Okay, so guys, no, ano yung purpose ng embedding natin? Lalagay natin yung ating tissue sa mold. Okay, so we're going to put our tissue in the mold. That is to produce now yung product natin na tinatawag nating tissue block. Tissue block. Okay? So guys, no, yan, tissue block. Yan. We have the tissue block. Okay? So guys, no, ano daw naman, okay, yung ginagamit nating ratio for the solution and tissue volume para sa infiltration. It's 25:1. Ibig sabihin, the volume of our paraffin wax or the infiltrating medium should be 25 times more than that of the volume of our tissue. Okay, so ulitin ko, for our fixation, it's 20:1. For dehydration and clearing, it's 10:1. Ibig sabihin, this is the fluid to tissue ratio. Fluid is 10 times the volume of our tissue. Sa infiltration, it's 25:1. Ibig sabihin, fluid is 25 times the volume of that of your or the fluid volume is 25 times the volume of your tissue. Okay? And then after that, we have the embedding or also known as your casting. Cast. Cast lang malakas. Joke. Ay casting. Hindi cast College of Allied Health Sciences. Eh di casting. Okay? Yan, casting or your embedding. Again, the medium used for embedding is also paraffin wax. Commonly used medium is paraffin wax. So tandaan ninyo, we usually use, or the medium that we use for embedding is the same as the medium, okay, we use sa infiltration or impregnation. So guys, no, here naman is a picture of a microtome. So this is where we do the cutting. So guys, napansin nyo, this one, okay, is actually the tissue block. And it's actually fitted dun sa ating tissue holder. And this one naman holds now our knife. Ito yung knife niya. Okay? And this one, guys, parang wheel ito. Ito yung handle niya. Iniikot-ikot natin yan. Pag inikot-ikot mo to, guys, no, bababa, tataas itong tissue block. At kakaskas siya doon sa ating knife. Kakaskasin ng knife natin. And magpo-produce tayo ngayon ng tissue ribbon. Okay? Yan, may mga impression siya nung tissue. Okay? So that's your cutting. Again, the most commonly used type of microtome is our rotary microtome, or also known as our minute microtome. Okay? So next, we have our next one here. Okay. So guys, no, I have here, okay, the link for tissue processing for light microscopy. So you can watch, okay, the processes, okay, done in preparing tissue slide, okay, or you may watch after, okay, the lecture, okay. So I have the link here. You just have to click it, okay. Okay, so next we have another method of histology is known as your autoradiography. Okay, so guys, no, your autoradiography is a method, okay, that we use in localizing newly synthesized macromolecules in cells or tissue sections. Like for example, we want to determine where in the cell, okay, these proteins or these glycoproteins are being produced. Okay, from the term autoradiography, okay, we use here a radioactive material. Okay? So guys, no, yung ating product for autoradiography is usually viewed under your transmission electron microscope. Bakit transmission? Kasi nakikita natin, 'di ba, yung inner, okay, a portion ng ating cell, for example. So we can appreciate where in the cell this macromolecule is being produced. Okay? And then we can also use your light microscope. Okay, we can use the light microscope for viewing. So we have here a picture. Okay, hold on. Ayan. Okay, so guys, no, this is a picture of the salivary gland of a mouse. And this salivary gland is injected with your 3H fucose 8H, okay, before the tissue fixation. Okay, so your fucose is incorporated into oligosaccharides. And the results now, okay, of this infusion is the revelation, okay, of the location where glycoproteins are synthesized. So saan napoproduce, saan sa tissue or sa cell, I should say, napoproduce yung mga glycoproteins natin. Okay? So guys, no, for letter A picture, we can see here the black silver grains. Makikita nyo yung mga dot-dot na black. Ayan, no, yung yan. Yan, mga black dyan. Okay? Yung mga black dyan. Okay? So yung mga regions na yon, okay, are actually regions with secretory granules. Okay, and that indicates now the location where glycoproteins or glycoproteins, I should say, are being synthesized. So, ibig sabihin, dito sa mga may black areas na to, dito sa part ng cell na ito, napoproduce or ng tissue na ito, napoproduce yung mga glycoproteins natin. Okay? So, this is under your transmission electron microscope. And then we have here another picture, letter B. Okay, so the same tissue is being prepared for your TEM. Okay, this one is your TEM. Okay, so makikita natin dito guys yung mga silver grains. Okay, so they come as coiled or amorphous. Okay, structures. So pwedeng ito, mga coiled appearance. Ayan o, coiled sila, di ba? Mga parang kulot-kulot na ganyan sila. Okay. Okay? O kaya naman amorphous. Ayan, parang mga maliliit na brick brick-like, okay? Mga powder-like, okay? Granules sila, okay? So, yan, same lang din dito, okay? So, sa letter A natin. So, nalolocate natin in what specific areas or locations, okay, in the cell, okay, are the macromolecules being synthesized, okay? So, that's because of your autoradiography. So this is again the application of your autoradiography. Okay, or it enables us to localize, okay, the location of the synthesis, okay, of certain macromolecules. So where in the cell are these macromolecules being produced? So nakikita natin sila using your autoradiography technique. So kamukha ito guys ha, nung parang x-ray ninyo. Parang ganun siya. Okay, yung principle niya. Okay? So next, we have your cell and tissue culture. So guys, no, another histology method is cell and tissue culture. Okay? So I believe you've heard about tissue culture and cell culture. Okay? Even in plants, they do that or they do this. Kaya nga kung may naririnig na kayo sa mga plantita natin, they have what is they call na tissue culture na plants. Okay, these plants were actually produced in the laboratory, okay, through a culture medium. Okay, so ganun din dito. So cells can be grown from a newly explanted tissue or as long established lines and can be examined in the living state by phase contrast light microscope. Okay? Guys, a very good example nito, may yung mga skin graft na kung naano nyo na yan, na narinig nyo na. Like for example, di ba pag nasunog, for example, yung muka, for example, or yung leeg ng isang pasyente natin, tapos gusto niyang mag-undergo ng plastic surgery, okay? They can do skin grafting, di ba? They get a skin or tissue, kunwari sa puwet, kasi yun yung hidden eh. So kahit tuklapan nila ng skin or kuhanan nila ng balat, hindi nakikita kasi hidden naman o kaya sa hita niya. Okay? And then they culture that and then they actually use that to grow new cells. Okay? Para yung term, okay, ano will how you call this one, yung layman term, parang itatapal nila dun sa areas, okay, na nasunog sa mukha or sa kanilang leeg. Okay, so parang ganun siya yung isang idea niya, tissue culture. Other than that, we can also use this in the study of your microorganisms. Halimbawa, yung mga viruses natin, di ba, we cannot study them in vitro. So, kailangan nandun sila sa mga tissues natin. So, we culture them in tissue in living cells, okay, yung ating mga virus. Okay? So, those are examples, okay, of applications of your cell and tissue culture. So, yan. Your cell culture has been widely used for the study of your normal and cancerous cells. So, paano sila nakaka-develop ng new drugs, paano sila nakaka-develop ng new tests, okay, na pwedeng gamitin dito sa mga actively multiplying cells na to. That's by studying them, okay, and culturing them. Okay, mamaya makikita nyo yung example din ng cell culture natin, guys, in the production of your antibodies. Okay? So, sa ano naman to, laboratory diagnostics naman yung application niya. Okay? And also, in microbiology, we have there. So, we can grow how do you call this one, microorganisms within cells, like for example viruses, mycoplasma, and some protozoa. Okay, so that's the purpose of your cell culture or your tissue culture. Okay, so next we have also your enzyme digestion. Okay, guys, now what's the purpose of your enzyme digestion? Andito siya. Okay, it is to confirm the identity of a stained material such as glycogen, DNA, and RNA. Guys, no, yung enzyme digestion, usually ginagamit natin ito for your nucleoproteins. So, we're going to detect, is it DNA, is it RNA? Okay, so we have here the applications. Okay, intracellular material that stains with fast reaction may be identified as glycogen by pretreatment of sections with diastase or your amylase. So with your deoxyribonuclease, okay, so paano ito? Okay, we have here an example. Okay, so guys, I'm discussing an overview of these methods. This is not an in-depth discussion, okay, because how you call this one, you will be discussing these dun sa mga higher subjects ninyo, okay? So this is for you to be provided of basic ideas, okay, about the methods, okay? So we have here, okay. We have here the process. Ano yung enzyme digestion na tinatawag? Okay, from the term itself, enzyme extraction or digestion, we remove something, okay, to confirm the presence of something, okay? So example, we have the DNA or enzyme extraction of the DNA, okay? So guys, now we're going to extract the DNA using an enzyme deoxyribonuclease, di ba? So if, for example, this is your cell, it has, for example, DNA, okay, in the nuclei, for example. So to confirm kung yung na-isolate ba natin na nucleoprotein is really DNA, okay, is we're going to perform, okay, an enzyme extraction procedure, okay? So we're going to remove or extract this DNA using now your deoxyribonuclease or yung tinatawag nating DNase. Okay? So the reagent is your deoxyribonuclease, 0.2 molar of tris buffer at the pH of 7.6 and distilled. So pag nag-apply tayo, okay, nung DNase ninyo or deoxyribonuclease, the DNA in this cell will be extracted. Tama? So mawawala siya dyan. That when you're going to stain this cell, okay, using your Fuelgen, okay, method, ang magiging result mo ay DNA negative. Bakit DNA negative? Kasi nga, na-extract na yung DNA na wala na si DNA. Okay? So that confirms na yung in-extract mo ay talagang DNA material, not RNA. Okay? By the way, ano ba yung Felgen technique or method? The Felgen method is a staining method that we use now to stain our DNA in tissues or cells. Okay? So it stains now the DNA red. Okay? So ulitin ko ha. This is the, hold on. Okay, this is the cell. Okay, so let's check if it has DNA. Okay, so we are going to extract this, okay, using your DNase. Okay, we've extracted this using your DNase. Kung DNA talaga siya, mawawala siya. Okay, absent na siya. That upon staining with your Felgen technique, there will be no material to be stained red. Bakit? Kasi si Fuelgen, it stains DNA red. Since na-extract si DNA mo, wala siyang i-stain. Kaya ang positive result dito ay DNA negative. Okay? The positive result is DNA negative. Ibig sabihin, yung extracted material mo ay DNA. Okay? Okay. So next, we have another technique. This is what we call naman your enzyme extraction for RNA. Okay. In this one, the staining technique that we use is your methyl green pyronine method. Your methyl green stains now your RNA, your methyl green stains your, hold on. Your methyl green stains your DNA green and your pyronine stains your RNA red. Okay? So guys, no example, this is your tissue or your cell. It contains both your DNA and your RNA. Okay? So guys, no, sabi niya, we're going to do enzyme extraction using your ribonuclease or yung tinatawag nating RNAs. RNAs. Okay? So, yan yung RNAs natin. So, pag tinanggal natin siya, ang matitira na lang dito, guys, ay DNA. Tama? And then, we're going to stain using your methyl green pyronine. Okay? Guys, pag nag-stain tayo with your methyl green pyronine, anong kulay yung lalabas? Ang kulay na lalabas ay for RNA, it's negative. For DNA, it's green. Okay? That confirms na na-extract natin si RNA. Okay, but of course, we have the control section. Okay? You have to get a tissue section that has both. That is to confirm naman kung talaga ba na na-extract si RNA o baka naman kasi talagang wala ng RNA dati doon. Okay? So yung control section mo dapat, it will give you a result of RNA in red color and DNA in green. Kung RNA negative din ito at DNA green siya, ibig sabihin, malamang yung test section mo, kaya nag-RNA negative siya, okay, is that talaga namang wala na siyang RNA. Okay? Kasi control section mo pa lang, negative na siya. Okay? So it's either, it gives an impression that it's either wala, how do you call this one? It's either sila yung how you call this one? Your RNAs, hindi mo madedetermine yung viability, okay, nung enzyme na ginamit mo kasi wala naman pala talagang RNAs. Okay? So, if for example naman, you extracted no using your RNAs. So we have for example this one, DNA, and then we have your RNA. Okay? So guys, no, for example, yan. Nag-extract ka ngayon using your ribonuclease or your RNAs. Okay? The enzyme RNAs. So, di ba yung RNAs mo, it will extract your RNA. So, ang result dapat niya, guys, si DNA lang yung matitira. Tama? DNA. Pero ang nangyari, guys, no, nung nag-stain ka with your methyl green pyronine, meron kang nakitang kulay na red at green. Okay, may nakita kang kulay na red at green. Ano ibig sabihin nun? Ay, hala. Na-stain ko, may DNA ako kasi merong green color, meron din akong RNA pala. Kasi merong red color. So anong pwedeng nangyari? Pwedeng yung RNAs mo hindi niya ginawa yung kanyang work. Pwedeng expired na yung reagent mo. Pwedeng your reagent is not viable anymore. So it's not capable anymore of extracting, okay, yung RNA mo. Kaya despite the application of your RNAs, nagkaroon ka pa rin ng positive result doon sa iyong test section ng RNA na red color. Okay? So, pwedeng hindi nag-work si RNAs mo kasi hindi niya na-extract si RNA mo, na-stain pa rin siya. Okay? Kasi kung nag-work sana siya, ang lalabas lang na kulay ay kulay green kasi si DNA lang yun, wala si RNA dahil na-extract siya. Okay? So, that's the enzyme extraction of RNA. Okay? And DNA. Okay? So guys, what's the purpose of your enzyme extraction or enzyme digestion? That is to confirm presence of your RNA or your DNA in your specimen. Okay? So next. Okay. Yes, okay. So this time we have your enzyme histochemistry. Okay? So guys, no, here we will be discussing naman, okay, yung um uh, method, okay, where we look into the activity of our enzymes. Kamukha ito guys ng autoradiography. Dito nga lang yung enzyme activity yung hinanap natin where in the tissue or where in the cell, okay, this enzyme, okay, doing its activity. So we do localization of the enzyme, okay, by detecting where exactly in that cell or in that tissue, this enzyme performing its activity, okay, or its function. Okay, so that's your histochemical techniques. Again, we have here the definition. It's used, okay, it uses specific enzymes activities in lightly or unfixed tissue section to produce visible products in specific enzyme locations. Okay? Again, we're not actually detecting yung enzyme mismo, but we're detecting where in the tissue it's doing, okay, its activities. Okay? Kasi ang dinedetect natin dito yung mga enzymatic products. Okay? So the tissue that we use here are frozen section tissue. So they are actually cut in a specialized microtome we call your cryostat. Your cryostat, guys, is also known as your freezing microtome. Actually, in freezing microtome, it's the, how do you call this one? The conventional, okay, counterpart ng cryostat. natin. Okay? It's actually like your rotary microtome inside a refrigerator. Para siyang nasa loob ng malamig na environment. Okay, bakit? Kasi in frozen tissue section, ang ginagamit natin ay fresh tissue. We do not fix it using yung routine techniques natin where we immerse it for 24 hours, for example, in your formalin. We do not do that here. Kasi nangyayari, nagsisira yung enzymes mo. So what we use if ay frozen section. Okay, instead of the tissue that has been processed with your routine histotechnics, yung mga diniscuss kanina natin na fixation, dehydration, hindi ganun. Okay? So ito frozen siya. So we get it from, for example, from the specimen of our patient fresh, then we immediately process it in a cryostat. Okay? So example of enzyme classes used here, we have your phosphatases, dehydrogenases, and your peroxidases. Okay? So guys, now we have here an example, okay, of your specimen, okay, processed histochemically. So kung makikita nyo, these are micrograph. In letter A, it's a micrograph section of the kidney tubules, okay? And it's stained using your Gomery method for your alkaline phosphatase. So kung makikita nyo guys, yung mga tinuturo ng arrows na to, these ones here, ibig sabihin, dito sa areas na to, okay, meron tayong activity nung alkaline phosphatase. Kamukha siya, kung maalala nyo kanina guys, yung autoradiography, di ba? We are actually localizing there to kung saan, okay, or where in the tissue or cell are the macromolecules being produced. Here, we're actually localizing, okay, enzyme activity. Okay, so this is for your alkaline phosphatase, and this picture shows now an image of the kidney tubules. Okay, and for letter B, we have here a TEM image of a kidney cell. Okay, and we are actually seeing now localizations of acid phosphatase activity. So dito daw sa areas na to. Okay, so ito yung nucleus niya guys. Nucleus niya to, ito yung cytoplasm, and these are your lysosomes. Itong mga bilog na to. So we can see here activities of your ACP or acid phosphatase. This is under your transmission electron, kaya mas malaki. Kita mo yung mga organelles. Okay? So, ayan siya. Okay? So, the dark materials within this structure is lead phosphate that precipitated in place with acid phosphatase activity. Okay? Again, we have localized where in the cell, okay, the enzyme is doing its activity, okay, by the precipitates produced, okay, by its products. Okay, yung product ng enzymatic reaction, okay, na kinakatalyze nung enzyme of interest natin. Okay, so ito sila. Dito siya nagpe-perform ng kanyang activity. Okay? So that's for your enzyme histochemistry. Okay? So this time, let's move on to the next. Wait, guys. Ayan. Okay. So we have here the application of our enzyme histochemistry. Example, the localization of the following. For example, no, your Pearl-Prussian blue reaction, okay, that detects now iron storage diseases. So saang part or dinedetect niya anong type ng enzyme yung merong missing ang activity. Okay, that's why meron tayong accumulation ng ganitong substance kasi wala yung enzyme necessary for its metabolism. Okay, kaya naiipon, okay, yung mga substance na ito. Okay, so let's detect what enzyme, okay, is not doing its activity, okay, or has decreased activity. Okay, also we have the past amylase and Alshan blue reactions. Okay, so this technique naman is for glycogen and glycosaminoglycans to detect glycogenosis and your mucopolysaccharidosis. So guys, no, you will no more of this pagdating nyo sa histopath, and some of the diseases will be introduced to you pagdating nyo naman sa clinical chemistry. Okay? And then reactions for lipids and sphingolipids. Okay? To detect naman sphingolipidosis. Okay? Like for example, bakit may ganitong may accumulation nitong substance na to? Okay. Kasi yung enzyme na to has a missing activity. So the enzyme is lacking, kaya itong precursor substance na to, hindi siya nakakonvert sa substance na to kasi wala yung enzyme na nagko-convert sa kanila. Hindi natin ma-detect yung kanyang activity using enzyme histochemistry. That's why this precursor substance, okay, are actually stored, okay, and are not metabolized. Okay, so nagkakaroon tayo ng accumulation, okay, nung substance na yon. Okay, so we detect that using that method. Okay, so enzyme histochemistry. Okay. So next, we have visualizing specific molecules. So guys, now in visualizing specific molecules, we have the following method. We have your immunohistochemistry and your hybridization technique. So in your immunohistochemistry, guys, we will be discussing two techniques, two basic principle. We have the direct and your indirect. Okay? Bakit? Na-introduce ko na to kanina sa inyo. Okay? Dun sa fluorescence assay, nung nandun tayo sa fluorescence microscopy. So guys, no, in your direct technique, we're just using a single antibody to detect presence of an antigen. In your indirect, we're actually using another antibody other than the primary antibody. We're using a secondary antibody to detect the binding of the primary antibody to our antigen. Okay? While in your hybridization techniques naman, it talks about now detecting, for example, an infecting microorganism using, okay, how do you call this one? Complementary nucleotide bases. So mamaya i-discuss natin ano ba yung mga pinagsasabi ko. Okay? So, ayan. So, let's move dun sa next slide. Okay? Hold on. Okay, so we will be discussing about immunohistochemistry first, okay? So when we say immunohistochemistry, okay, it's based on specific reactions between antigen or antibodies, okay? So when we say immuno, we are actually talking about your antigen antibodies, okay? Histo, we have tissue, chemistry. Okay? So, yan. Sabi niya, between an antigen or antibodies labeled with visible markers. Okay? Often fluorescent compounds. Maalala nyo kanina guys, dun sa sa direct fluorescence assay, di ba? Yung ano natin, fluorochromes natin, binigyan ko kayo ng example. We have your fluorescent isothiocyanate, your auramine, your rhodamine. Okay, so these are examples of markers, okay, that we use. But you have to take note na hindi lang fluorochromes yung ginagamit natin, guys, na label for our antibodies. We may use particles like latex, okay? We may use proteins, okay? We can also use example yung biotin avidin ninyo, okay, to label, okay? So, or enzymes, okay? So, depende. And of course, the reading, okay, of the result depends now on the label that you use. So if you, for example, use no fluorochrome, then you can use the result using your fluorescence microscope because the result will be in fluorescence. Okay. So if, for example, you have used an enzyme label, so the result, okay, can be read using a colorimetric assay. Okay. So you can determine the result or the result is indicated by a color change. Okay? So, yun yung mga examples natin. If, for example, the antibody is labeled with your radioactive particles like your iodine 125, for example, then you can use techniques like radio immunoassay, okay, to read the label. Or we call the technique as your radio immunoassay. Okay, if you're using a fluorochrome, then that's a fluorescent assay. Okay, if you're using an enzyme, then that's an enzyme assay. Okay? Or a colorimetric assay. Okay? So, yan. So, guys, no, in your immunohistochemistry, sabi natin, we're talking about your antigen and antibodies. And as I mentioned a while ago, we have two basic techniques. We have your direct and your indirect. So, tandaan ninyo, in your direct immunohistochemistry, we can detect presence of an antigen through the labeled primary antibody. So yung label dito is yung primary antibody. And actually, isang antibody lang ang ginagamit natin. Kasi if this is the tissue, for example, antibody type, I should say, okay? Ito lang yung antibody. We need not other antibody to bind this antibody. Siya lang. If this is yung example natin kanina guys sa direct fluorescence assay, di ba? Meron siyang antigen. May rabies antigen dito sa tissue natin. Tapos, meron tayong reagent antibody. So, i-confirm natin, mayroon ba talagang rabies antigen yung tissue? So, we will use, okay, we're going to use, okay, a reagent antibody. And this reagent antibody is known as your anti-rabies antibody. So, kapag nilagay natin yung ating reagent antibody, okay, and then we incubated it with our specimen, magbabind ngayon si reagent antibody with our antigen. Okay? Nuwari, fluorochrome labeled siya. Okay? So, we are going to read, this is a fluorescence assay, so we'll read the result under your fluorescence microscope. So, kung nagbind siya, makakakita ka, for example, kung fits yung ginamit mo, we have what? Apple green fluorescence. Okay? So, isa lang yung antibody, primary antibody. In your indirect, it's the secondary antibody that is labeled. Paano naman, ma'am, yung sa indirect? Guys, no, if for example, this is the tissue, sorry, the slide. We have here, no, the fixed tissue in the slide. For example, this is your antigen A. Okay, antigen A. Okay, guys, no, we are going to use two types of reagent antibody. The primary antibody here is known as your anti-A antibody. Bakit? Kasi guys, itong anti-A antibody na to ay gagamitin natin to detect kung meron ba talagang antigen A si tissue natin. Okay? So ito si antigen A, for example, dun sa tissue. So pwedeng absent o pwedeng meron. So halimbawa, meron. Okay? So we can detect their presence, okay, or the presence of these antigens by using now a primary antibody against antigen A. So again, these antibodies are your anti-A antibodies. Okay? And then, other than using, kasi kung mapapansin nyo guys, no, yung primary antibody natin sa indirect, hindi sila labeled. So kahit mag-bind ngayon si antibody kay antigen, pwedeng hindi natin mabasa yung result. Okay? So ang gagawin natin, to intensify, okay, the result, we use a secondary antibody. Okay, gagamit tayo ngayon ng secondary antibody. So this one is the secondary antibody. Guys, no, ano naman yung secondary reagent antibody natin? Guys, no, yung secondary reagent antibody natin, sila yung label. Example, fluorochrome. Ang label natin, halimbawa lang ha. Okay, o yan. Yun, itong mga antibodies na to, ma'am. Ma'am, itong mga antibodies na ba to ay anti-A antigen antibody din sila? Hindi. Okay? These antibodies naman are directed against your primary antibodies. Okay? So, ibig sabihin, these are your anti-A, these are your anti-human globulin or AHG, or they are actually ano ba tong antibody na to? Di ba, these are your anti-A antibodies. So this one are your anti-A antibodies. Ibig sabihin, they are against, or they are against your anti-A antibodies. Okay? Nakuha nyo ba? Okay? So ulitin ko guys ha. These primary antibodies are directed against your antigen. Okay? These primary antibodies are directed against your antigen of interest. If this is, for example, antigen A, then these primary antibodies are your anti-A antibodies. Okay? Your secondary antibody naman, on the other hand, is directed naman against your primary antibody. So they are your anti-primary antibody, or sometimes they can also be called as your anti-A antibodies. Okay? So anti sila dun sa anti-A antibodies, or we can call them as your AHG or anti-human globulin. Okay? So secondary antibodies are directed against your primary antibodies. Ibig sabihin, ang nagiging antigen ngayon ni secondary antibody are your primary antibodies. Okay? While your primary antibodies are against naman the antigen found in your tissue. So tatandaan nyo, in your indirect, we have two types of antibodies used. We have the primary and the secondary. Guys, sino yung merong label? It's the secondary antibodies. Okay, so guys, no, si secondary antibody yung may label. So paano ngayon yan? Okay, so ganito yung magiging itsura nila kapag ka present yung antigen natin sa tissue. So hold on. We're going to, so this is now your slide and this is your tissue. Okay? Guys, no, meron tayo nung antigen A. Ilalagay natin yung primary antibody natin. Si primary antibody, walang label. Tama? So, ibabind niya ngayon, your primary antibody is your anti-A antibody. So, it will bind now the antigen. And then after that, we're going to rinse it, wash it. That's to remove no excess primary antibody, yung mga unbound. Siyempre kung unbound sila, hindi sila din nakafix sa cell kasi hindi sila nakadikit dun sa mga antigen. So for example, primary antibody. So dahil dadalawa lang yan, kunwari meron pa tayong isang primary dyan. O, so yung mga unbound, ito unbound siya, wala na siyang pagkakapitan na antigen. Pag nag-rinse tayo or nag-wash tayo guys, itong antibody na ito mawawash out. Okay? Mawawash out siya. So tatanggalin natin siya kasi nag-wash tayo. Okay? So ang matitira lang yung mga nakabind dun sa antigen natin. Kung dalawa yan, so may dalawa din tayo nung antibody. Then we will detect, okay, presence of this binding or reaction. This is an antibody antigen complex, right? So we will detect the presence of these, okay, complex, okay, by using now another antibody. And that's naman your secondary antibody. Again, your secondary antibodies, they are labeled. Other than that, they are directed against your primary antibody. Hindi sila magbabind dun sa antigen mo because they are not anti-A antigen. They are actually anti-A antibody. Okay? So against sila dun sa antibody. Okay, na ginamit mo against your antigen sa tissue. So they will bind now, okay, dun sa ating antigen, dun sa kanilang Fab region. So guys, no, if this is your antibody pala, ito yung tinatawag natin guys na Fab region. Okay, ito, yung parang pinakaulo niya. This is the Fab region or the fragment antibody binding. Hold on. Nawala tuloy. Oh, so this is the antigen, okay? This one here, okay? Or ito, this is your Fab or yung tinatawag nating fragment antibody binding. And this one naman here is your fragment or your FC region or also known as your fragment crystallizable. Okay? So ayan, ulitin natin na. Ito yung tissue natin. Ito yung, yan, meron siyang antigens. Ngayon, nakabind na sa kanya yung primary antibodies natin. Okay, nakabind na. Ngayon, nagdagdag tayo nung secondary antibodies. Guys, yung secondary antibodies against natin, okay, ay labeled. Saan magbabind saan magbabind or saan ibabind ni secondary antibody si primary antibody? Dito, sa kanyang Fab region, sa fragment antibody or fragment antigen binding. Okay, right? Kasi si antibody na ito, itong antibody na ito ay kanyang antigen, right? Okay? So it will bind it this way. Okay? So pwedeng hold on. Yan. So pwedeng kakabit yung isa diyan. So this is the fragment, right? the fragment antigen binding. Okay? So, kakabit siya dyan. Pwedeng another din, kakabit isa dyan. Okay? Pwedeng another ulit, dito ulit. Kakabit ulit yung isa dyan. Kasi directed sila against these antigens. Okay? So, guys, no, labeled sila, right? So, anong napansin ninyo, guys? Okay? Hold on, ah. Wait lang, I will make it larger. Ano ba? How is it? Okay. Hold on. Yan. I will draw. Okay, yan. And then, yan. Ay, red din yung kulay. Okay. Oh, so guys, ito siya, di ba? Yung tissue slide natin. Yan. Ito yung antigen ulit. Sorry ulilit ko lang kasi para makita. Ayan. O, di ba? Yan na yung antibody natin. Tapos ito yung isang antibody. So, ito na yung secondary antibody labeled. Yung isa, pwedeng may kumabit dito. Okay? Pwede namang may kumabit din dito, guys. O, labeled siya. Okay? So, pwede din namang meron dito, kakabit na antibody. So, labeled yan. Secondary antibodies lahat to, yung kumabit dun sa mga primary antibodies natin. Okay? So, guys, napansin niyo dun sa mga primary antibodies natin, madaming pwedeng kumabit na secondary antibodies. This is your indirect. Ibig sabihin, guys, look at that. It's very sensitive. Okay? That even in a small amount of antigen, kaya niyang i-amplify yung result mo. Positive result. Kasi madaming primary antibody ang pwedeng mag-amplify ng result. Bakit sila yung labeled, di ba? So, pwedeng madaming antibodies yung kakabit dun sa primary antibodies natin. We're referring to the labeled secondary. Unlike dito sa direct, maalala nyo yung direct, if this is the specimen, pareho tayo merong dalawang antigen. Di ba, nade-detect natin yung presence ng antigen by using only a single antibody and that's your primary antibody. Siya yung labeled. Yan. Okay? So, imaginein mo yung two signals na binibigay nitong direct technique mo compared dito sa two, four. Four na signals na binibigay ng indirect technique. Okay? So, anong ibig sabihin nun, guys? Your indirect technique, okay? Indirect. This is your indirect and this is your direct. Your indirect technique is highly sensitive, okay? Compared to your direct technique. Isipin mo yon, pareho sila dalawang antigen, pero yung intensity ng result or indication ng positive result, mas intense dito kay indirect. Kasi kung mapapansin mo, meron ka na ngayong total of 4 or pwedeng madami pang kumabit dyan, okay, na secondary antibodies. Okay? So, madaming fluorochrome labels kang makikita. Unlike here, dalawa yung antigen mo, so dalawa lang yung antibody that will bind it kasi isa lang yung ginamit mo. You're using the direct technique. Okay? So that's the difference between your indirect and your direct immunohistochemistry. So mamaya makikita natin, guys, what's the difference. Okay, other than of course your indirect technique is more sensitive than that of the direct technique. Okay? So guys, no, kanina, di ba, in the detection of your antigens, we're using antibodies. Guys, no, the antibodies that we are using in immunohistochemistry or in your serologic techniques, okay, are either polyclonal antibodies or monoclonal antibodies. Anong ibig sabihin? When we say when we say polyclonal antibodies, these are mixture of heterogeneous antibodies. Anong ibig sabihin? Okay, so these are mixtures of heterogeneous antibodies that are usually produced by different B-cell clones in the immunized animal. Okay? Ano ba yung ibig sabihin? Mamaya di-discuss natin. Okay? Bakit yung mga antibodies na yun naging heterogeneous? Kasi they're coming from different B cells. While your monoclonal antibodies naman, they are from identical B cells. Identical B cells. So, ibig sabihin, iisa lang yung clone na pinanggalingan nila. Kunwari, parang ganito siya. Um, Paano ko ba i-explain? For example, eto si okay, kunwari ito, sa polyclonal, meron tayong mga different antibodies. May, B cell, kako sana. Merong B cell na red, merong B cell na green, merong B cell na pink. In your monoclonal, na-isolate sila. Kunwari, ang B cells natin, puro orange sila. Okay? So, if they're going to produce antibodies, itong mga orange B cells natin, puro orange. Kasi pare-pareho sila, puro orange na B cell. Guys, the color, I'm just using this for to demonstrate to you, okay, in simple way, kung ano yung ibig sabihin ng monoclonal antibodies produced in identical B cells and polyclonal antibodies produced in different B cells. So, for these naman B cells here, example, this one here, this red B cell will produce red antibodies, right? And then this pink B cell will produce pink antibodies. And this green B cell will produce green antibodies, okay? So, napansin ninyo, okay? For example, we're going to use these antibodies in the detection of of certain diseases, okay? Of a certain disease. Okay? So, pwede natin gamitin lahat to ng antibodies na to. At ganun din itong mga antibody na to. Okay? So, halimbawa, we have this antigen. Okay? For example, an infectious disease. Meron tayong antigen. Ito guys, kunwari yung antigen natin. Okay? So, meron tayong antigen. Ito siya. Guys, no, yung antigen natin, it has what we call these epitope. Okay, yung mga active sites ng antigen natin. Okay, so this is where actually your antibodies are binding the antigen dun sa epitope nila. Okay, so epitope. So you will know more of this pagdating nyo sa immunology and serology. So guys, no, yung antibody na to ay meron siyang iba-ibang epitope. Okay, meron siyang hold on. Meron siyang pink. Ay, sorry. Meron siyang pink na epitope na bilog. Okay? Meron din naman siyang red na epitope. Okay? Nakuha niyo ba ako? Meron din naman siyang green na epitope. Yan. O meron din naman siyang orange na epitope. Okay? For example, like that. Okay. Okay? So ito yung antigen natin. It has different epitopes, okay, on its surface, okay? And it is on the epitopes where your antibodies bind, okay? Guys, no, siyempre, mapapansin ninyo, if we are using polyclonal antibodies, anong ibig sabihin nito? Yung antibody natin pwedeng produce from a B cell, okay, that is pink color, pink din siya. Kung green siya, di green din yung antibodies niya. Yung yellow siya, yellow din. So this B cell here, okay, producing an orange antibody will have an antibody, orange ba yun? Sorry guys, red, okay? Will have an antibody directed against the red epitope. Okay? Gets nyo na? Okay, dito siya. So, ito ay antibody na produce ni red. Now, if we have also kasi polyclonal antibodies yung ginamit natin, so pwede iba-iba sila, okay, na population of antibodies. So, we have also here the green antibody. Okay? So, guys, no, yung green antibody, ibabind niya naman si epitope na kilala niya. Okay? Kilala niya. Okay? So, the green antibody will bind the green antigen. Ibig sabihin, si green antibody ay anti-green antibody or anti-green epitope antibody. Nakuha? And then, we also have here si pink. O, si pink antibody, it is anti-pink antibody. So, it will bind the pink. Okay? So, nakita nyo, yung ating polyclonal antibodies, Okay? Since they are coming from different B cell, they are heterogeneous. Which means, okay, that their binding or they can bind, okay, with any types of epitope found in your antigen. Okay? Anong ibig sabihin nun? Mamaya, ito naman. Okay, dito muna tayo sa polyclonal. Okay? Sa monoclonal, I should say. So, we have the same antigen, okay, or microorganism. So, meron siya ng epitope, pa. So, meron din siya nung epitope na pink, guys. Meron siya nung epitope na orange or na red, sorry. Meron siya nung epitope na red. Same lang dun sa isa, gayahin natin yung isa kasi pareho sila for example. Okay, and then we also have that green epitope. Okay, and then we also have orange. Okay, so madami siyang orange. Okay, so guys ha, tandaan ninyo, dito kanina, these antibodies here are your polyclonal antibodies. Okay, so they bind different types of epitope kung saan sila. So kung si pink, siya ay anti-pink. Hindi siya pwedeng maging anti-green. Nakukuha? Kasi pink siya. It's directed against the pink antigen. Okay? Guys, the colors, okay, I'm using here are just codes for you to understand that the binding, okay, or the relationship, okay, of these antibodies to their antigen. Okay? So, yan. So these are your polyclonal antibodies. If for example, dito naman ang ginamit natin ay monoclonal, guys, no, since pare-pareho sila, these antibodies, you will just see binding dito sa may orange. Kasi orange yung antibodies mo. I'm referring to this, hindi ko kasi napalitan yung kulay. Hold on. Ay, sorry. Okay, this one. This we have your monoclonal antibodies, right? So they will bind now in this epitope. Kasi orange sila. Okay? So yung monoclonal antibodies natin lang, iisa lang, identical. Okay? They are identical coming from a single parent. So si parent cell nila ay si B cell na orange. So magpo-produce lang siya ng puro orange. Ibig sabihin, if you're using a monoclonal antibodies, specific yung binding nila sa orange lang. Okay? Bakit? Kasi they're coming from a single parent cell. Orange lang sila. Anak lang sila. Hindi anak, sorry. They're produced by a orange B cell. Pag monoclonal, anong nangyari dun sa monoclonal, guys? Yeah, sorry. Ulitin ko ha. Monoclonal. This is your monoclonal. Okay? Ito yung monoclonal nyo, guys. Okay? Monoclonal, ibig sabihin, the cells where we derive the antibodies, okay, are clones of single parent cell. Identical sila. Okay? Kaya puro orange yung antibodies na na-produce. So if you can see, the antibodies are not binding any other epitopes except the orange epitope. Ito sila. Okay, right? So that's monoclonal antibodies. Here, in this box here, dito naman, you can see we have the polyclonal antibodies. The polyclonal antibodies are heterogeneous. They are produced by different B cells. Thus, they can be antibodies to different types of epitopes. Okay, in, for example, an antigen. Okay, so if you can see this antigen here, it has different types of epitopes. Okay? At yung nagbabind sa kanila na antibody are antibodies that are directed, okay, specifically to that epitope. Your pink antibody cannot bind the green epitope, right? Your red cannot bind the pink. So it will just bind the red. So with the green, it will just bind the green. Okay? So polyclonal. Okay? If you can see, the polyclonal antibodies, okay, are capable of binding the antigen but at the different portions or at different types of epitopes. Depende kung saan sila directed or against. But in your monoclonal, dahil isa lang na klase ng population ang napoproduce natin, puro orange, ibig sabihin, yung binding mo, nasa orange epitope lang. So guys, anong implication nito? Okay? Guys, no, the use of your polyclonal antibodies, okay, is what? Less specific than your monoclonal antibodies. Okay? So ulitin ko, your use of polyclonal antibodies, okay, makes the test non-specific or less specific. Okay? While your polyclonal antibodies, it's what? More specific. Diba? More specific. Pero tandaan ninyo, here, almost all of the epitopes, okay, dito sa use ng polyclonal natin, guys, halos lahat sila ng epitopes natin na bind, right? Kasi meron tayong iba't ibang uri ng antibody against each type of epitope. Dito, guys, napansin niyo dalawang epitope lang yung merong bound antibody. Bakit? Kasi dalawa lang sila na kulay orange. Dahil gumamit tayo ng monoclonal antibody, identical lahat ng antibody natin. They are directed against epitope orange. Dalawa lang sila. Okay? So ano naman ang implication nun? Ibig sabihin, guys, the use of your polyclonal antibodies, okay, will make the test more sensitive, and for your monoclonal, it's less sensitive. Okay? Ulitin ko. When you're using your polyclonal antibodies, okay, since iba-ibang antibody yan, guys, ibig sabihin iba-ibang epitopes pwede silang mag-bind doon sa ating antigen, then that will make the test more sensitive. Okay? While in your polyclonal antibodies, dahil single population of antibodies lang siya, directed lang sila sa iisang type ng epitope doon sa antigen mo, so ang ibabind lang nila ay yung epitope na nag-iisa kung saan sila directed. That, okay, is actually making the test less sensitive. Okay? But, since nag-iisang anti-epitope lang yung binabind nila dahil sila ay puro pare-pareho or sila ay identical, that makes, okay, the test more specific. Okay? But for polyclonal, dahil iba-iba silang types ng antibody against different types of epitope, ibig sabihin, kahit saan dun sa antigen, pwede sila mag-bind. That makes the test less specific. Okay? So, that's the difference between the use of your polyclonal antibody and your monoclonal antibody. Okay? So, mamaya i-review natin yan, guys. Okay? Ulit. So, next is yan. So guys, no, this is how your monoclonal antibodies and polyclonal antibodies are being produced. Okay, so this first picture here, guys, no, we have the production of your monoclonal, polyclonal antibodies. Okay, so guys, no, we're going to inject, okay, the animal or the laboratory animal with an antigen. Okay, and of course, this laboratory animal, okay, will actually, or the B cells, I should say, of this laboratory animal will be activated by the antigen, okay, na inintroduce natin. So if you can see here, ito yung ating mga B cells, okay? So these B cells are activated kasi nakabind na sila dun sa ating antibody. Okay? Ulitin ko ha, this is for your polyclonal. Okay? Guys, napansin, hold on. Okay. So guys, napansin ninyo ha, pansinin ninyo, this B cell here is producing an antibody na nagbabind or is against sa pink na antigen. Ayan yung pink antigen. While this B cell here naman adding ay nagpo-produce ng antibody naman na directed against the green epitope. Nakita nyo? Ito pink epitope, ito green. Same antigen, pero magkaiba yung binding site nila or yung epitope na binabind. Okay? So these are your, how do you call this one? These are your different B cells. Okay? So ngayon, these B cells will produce now polyclonal antibodies. Okay? So again, ano yung mga na-produce natin, guys? Okay? Na mga antibodies natin, polyclonal antibodies. Anong ibig sabihin? pwedeng yung antibody na ito ay directed sa epitope na pink. Ayan, dito. O kaya siya ay directed sa epitope na green, dito. O kaya siya ay directed sa epitope na kulay yellow. Okay? That's the production or how your polyclonal antibodies are produced. Okay? So ano yung fate pala no guys ng B cell mo? Okay? The activated B cells can become your memory B cells. Okay? Ito sila. Ano memory B cells? May memory sila guys. Why? Mamaya explain ko sa inyo yan. Okay? Or your plasma B cells. And these plasma B cells are the one actually producing your antibodies. Okay? So sila yung hina-harvest natin coming from this laboratory animal. At yun yung gagamitin natin as reagent antibody. So meron na tayong polyclonal antibodies. Bakit polyclonal? Kasi galing sila sa different population ng B cell. Pwedeng yung B cell na yon ay nagpo-produce ng antibody directed against the pink antigen, yellow antigen, o kaya naman green antigen. So polyclonal. Okay, heterogeneous yung antibodies mo. Okay? So guys, no, ano yung naging fate? O ngayon, discuss naman muna natin to bago tayo lumipat dun sa how you call this one? Sa, uh, tawag dito? Sa monoclonal. Okay? So yung fate, no, upon activation, your B cell become your memory B cells or the antibody producing cells, plasma cells or your plasma B cells. Guys, ano yung memory B cells? Saan to? Ano yung purpose nila to? Or what's the medical significance or clinical significance? Okay? So guys, no, like for example, 'di ba, kapag ma nag-measles ka na, 'di ba? For example, nag nagkaroon ka ng paano nga ba 'to? Nagkaroon ka ng measles, for example. So your body will, the antigen, okay, inhaled, or the virus, for example, will cause now the activation of your B cells. So it will produce antibodies, okay, in your plasma B cells, will produce antibodies against, guys, hold on, wait lang burahin ko lang kasi to make myself clear. Hold on. Ayan. So guys, ganito siya. So, 'di ba nagkaroon ka ng measles? Okay? So yung measles mo, it will activate your B cells. Magkakaroon ka ng sakit, of course, magkakaroon ka ng sakit. Okay? But of course, yung measles mo, okay, na nag-activate ng B cells mo, some of the B cells will turn into your memory B cells. Ibig sabihin, may memory sila. Okay? Okay, I have already met measles. Na-meet ko na tong si measles antigen. Okay, so yon yung memory B cells mo. Ano ibig sabihin? The next time na maka-encounter ka ng patient, for example, na measles, tapos na-inhale mo ulit, okay, di ba? Through inhalation ang mode of transmission ng measles, okay? So, na-inhale mo ulit, for example, okay, yung virus. Ang susunod, hindi ka na guys magkakaroon ng sakit kasi nagkaroon ka na ng sakit dati, na-activate na dati si B cells mo. So ano mangyayari? Meron ka na dati, yung mga memory B cells mo, naalala na nila dati si measles kasi nagkaroon ka na dati nung sakit. Okay? So mangyayari, they will just be activated again to become your plasma B cells and produce now antibodies against the antigen or the measles antigen. So sa susunod na encounter mo, hindi ka na ulit magkakasakit kasi you've already formed antibodies against the antigen that was introduced to you before, okay, nung nagkasakit ka. Okay? So yan, it has something to do with your adaptive immunity. Okay? So ayan, kaya hindi tayo nagkakasakit ulit or nagkakaroon ulit ng measles. Okay? So that's your memory B cells. Okay? So yan. Again, that's the purpose of your memory B cells na memorize niya, okay, yung mga antigens na na-introduce na sa'yo dati. And when you will be introduced again of the same antigen, it will be activated to produce antibodies against that antigen. Di ka na magkakasakit kasi nagkasakit ka na. We are talking about natural immunity ha. Okay? So, meron ka na nung antibody against it. Okay? So, hindi ka na magkakasakit. Okay? So, I'm talking about human. Etong nandito sa picture, guys, this is the production, okay, of your monoclonal antibodies naman in your animal or laboratory animal. I'm just explaining to you now the adaptive immunity kasi baka magtanong kayo, ma'am, ano yung memory B cell? Ano yung purpose nila or function nila? Okay? So, yan. It's for immunologic memory. The next time you will meet again the same antigen, maaalala ng katawan mo, oh, I've already met measles antigen. So, wow, I will produce antibodies against it. Hindi na ulit ako magkakasakit kasi I have already the antibodies against it. Okay? So, but again, wag kayong malilito. I'm just discussing that, the adaptive immunity or the natural immunity dahil dito sa memory B cell natin. Okay, kung ano function nila for example sa katawan natin as humans. Okay, but ang focus natin guys sa itong production ng ating polyclonal antibodies. Okay? So ulitin ko guys, yung mga na-produce natin na mga antibodies ay polyclonal, they are heterogeneous because they're coming from B cells that are actually producing antibodies directed against either the pink epitope, the yellow epitope, or the green epitope. Okay? So next is we have here, sorry, naman guys, yung inyong monoclonal. Okay? So sa monoclonal antibody, guys, medyo mas laborious yung procedure, okay, ng production. So we're going to immunize, okay, this rat here, the laboratory animal. Then after that, we isolate the B cell. Okay? So these are the antibody forming cells. Guys, no, we are going, okay, to get the B cell and fuse it. Okay? If you fuse natin yung B cell with a tumor cell. The tumor cell that we usually use in producing this product cell, which is called as your hybridoma, are your myeloma cells or myeloma tumor cells. Okay? Guys, ano yung purpose? Ma'am, bakit kailangan nating i-fuse itong antibody forming cell na to sa tumor cell? Okay? It's actually to make, of course nga, yung tinatawag nating hybridoma. Eh ano ba yung characteristic ng hybridoma? The characteristics of your hybridomas, okay, are actually the combination of the characteristic of an antibody forming cell and a tumor cell. Ano ba yung characteristic nitong dalawa? Guys, no, of course, the characteristic of your antibody forming cell is that it's capable of producing antibodies, right? So, etong hybridoma mo na to, ibig sabihin, magpo-produce din siya ng antibody kasi siya ay fusion ni antibody forming cell at tumor cell. Okay? Nakakapag-produce siya ng antibody kasi nga, it's actually coming from antibody forming cell fused with a tumor cell. Ngayon, what's the purpose? Ma'am, kasi di ba, why do you have to fuse it then with your tumor cell if your purpose only is to let it produce antibody? We fuse it with your tumor cell because, okay, of the purpose, okay, because of the purpose that your tumor cells has the following or tumor cells have the following characteristic. Ano yun? We have longevity and reproductivity. Guys, yung tumor cells natin, they are long-lived cells. Okay? They are immortal. Okay? So, unlike kasi pag solo antibody forming cell lang siya, after some time, okay, they will stop producing. Bakit? Kasi they will die. Okay? But when you fuse them with your tumor cells, okay, to produce this hybridoma, other than magpo-produce siya ng antibody, it also has this characteristic of longevity and reproductivity. Di ba ang tumor cells, they have this high mitotic activity. So, they have this ability to replicate, okay, and multiply a lot. Okay? So yun yung purpose natin to create this hybrid in creating this hybridoma. Why we fuse the antibody forming cell with your tumor cell. Again, that's to combine the characteristic of your antibody forming cell, hold on, which is wait, okay, which is to produce to produce antibody plus, okay, the characteristics, okay, of your tumor cells, which is longevity and reproductivity. Okay? productivity. Bakit? Kasi nga sabi natin guys, yung tumor cells ninyo, they are long-lived cells, okay? They have this ability to multiply a lot or do rapid multiplication. Okay? So, yan. I-combine natin yung mga characteristics nila to produce now yung tinatawag nating hybridoma. Okay? So, guys, no, the hybridomas will be screened for the production of desired antibody. Adding, yung mga hybridomas natin, ganun din sila, guys. Pag nag-multiply na sila, pwede silang directed against antibody epitope orange, epitope green, or epitope pink. Pero in your monoclonal antibody production kasi guys, meron silang tinatawag na antibody B cell isolation. Okay? So, ina-isolate nila. Aling mga B cells lang ang nagpo-produce ng antibody against, for example, epitope orange. Yun lang yung kukunin natin. So, i-ano nila, i-separate nila yung mga B cells na magpo-produce ng antibody against green epitope, dito kayo. Yung mga B cells na magpo-produce ng antibody against orange epitope, dito kayo. Yung mga magpo-produce against green epitope, dito kayo. So, isa-separate. Okay? So, they will just choose one. Okay, to make it more specific, okay, I will choose antibodies that will be directed against your epitope green. And yun yung i-undergo nila ng clonal expansion. Okay, and eventually, okay, it will cause now the production or it will lead to the production of your monoclonal antibodies. So maging yung monoclonal antibodies mo na ngayon, these are antibodies na directed lang sa single type of epitope. Okay, so monoclonal antibodies. Okay, so let's have a review. So guys, ulitin ko ha, dito sa polyclonal antibody production, heterogeneous yung antibody na napoproduce natin. Bakit? Kasi galing sila sa mga iba't ibang uri ng B cell. Pwedeng si B cell producing antibody against the green epitope or the pink epitope. Okay? Kaya yung napoproduce na antibody mo, polyclonal, kasi these are antibodies directed against different types of epitope in an antigen. Okay? Next, in your monoclonal antibody production naman guys, okay, before discussing yung type ng antibody na napoproduce, it is something laborious compared to your polyclonal antibody production. There's a need of fusing now an antibody forming cell with tumor cell. Bakit? That is to produce your hybridoma. Your hybridoma has characteristics, okay, that are combination of your antibody forming cell characteristic and that of your tumor cells characteristics. So other than it producing antibodies, okay, they have this longevity and reproductivity. Okay, so in the hybridomas, after that, these hybridomas will be screened. Ibig sabihin, there is this B cell isolation that we do. We separate B cells according to the type of antibodies they produce. If this B cell produce an antibody or antibodies against orange epitope, will isolate them from any other antibodies that produce, we isolate them from B cells that produce antibodies against any other epitopes. Like we separate B cells that produce antibodies against epitope green, against epitope pink. We separate also the B cells that produce antibodies that are against epitope red. Okay? So, ganun siya. Meron tayong isolation. So, we just have to pick one. Okay? Group of B cell population, identical B cell population, at yun yung i-undergo natin ng clonal expansion. Thus, the production of your monoclonal antibody. So if, for example, okay, we are going to do clonal expansion of B cells that are producing antibodies against the orange epitope. So, yung mapoproduce mo na ngayon, okay, na reagent antibodies mo, are more specific. They are for the orange epitope. Okay? So, that's monoclonal antibody production. Okay? So, guys, no, yeah. Hold on. Ito, hindi ko na to, kasi na-explain ko na siya kanina, yung difference ng polyclonal antibody at saka monoclonal antibody. Okay? So, ito lang siya, guys. Okay? I will just have to write this one. So, ayan. So, kung makikita nyo, this is polyclonal anti-serum. We have here polyclonal antibodies. So, kung mapapansin nyo guys, the antibodies are directed against different epitopes in the antigen. So makikita nyo, merong antibodies for pink antigen, for green antigen, for how you call this one? Yeah, for green and pink antigen. Okay? Dito sa monoclonal, pare-pareho sila. Silang lahat ng antibodies natin, nagbabind lang sila dun sa pink antigen. Sorry, pink epitope, I should say. Okay? Na portion ng ating antigen. Okay? Again, in your polyclonal, the antibodies are binding in the different epitopes in an antigen. So, pwedeng si pink epitope or green epitope. Sa monoclonal, single epitope yung kanilang binding. Kung pink silang lahat, pink lahat yan ang binding nila. Okay? So that's your monoclonal antibodies versus your polyclonal antibodies. Okay? So next, okay. So guys, ito yung process dun sa monoclonal antibody production. Okay? So 'di ba, meron dun sa monoclonal antibody, kunwari ang gusto nating paramihin or i-undergo ng polyclonal expansion is yung antibody against epitope A. Okay? Against epitope A. So ito sila. Okay? So, we have antibody against epitope P, epitope C. So, dun sa ano natin, sa hybridomas natin, pwedeng may B cell na nagpo-produce ng antibody B. Ibig sabihin siya yung against kay epitope B. Meron naman yung B cells na nagpo-produce ng antibody against epitope C. So, yun si antibody C. Yan siya. Or may B cell naman na nagpo-produce ng mga antibodies against epitope A. Siya si antibody A. So, against siya kay epitope A. Ito. Okay? So, yan. Ngayon, they will do isolation. So, kung makikita nyo dito sa picture, guys, they will put in microtiter wells yung mga B cell natin. So, this B cell here, this well here contains now the B cell that produce anti antibody A only. This one naman here contains now B cell that produce the green one. So that's ano yung kulay, ano yung green? Antibody B. And this well here contains now the B cell that contains naman the red antibody C. Okay? So i-isolate natin sila. Okay? Then they will choose which of these, okay, will undergo clonal expansion. So kunwari si B cell, yung mga B cells na nagpo-produce ng antibody against epitope green or yung epitope B natin, sila yung pararamihin natin. Okay? So that's your monoclonal antibody production. Okay? So guys, now we have here the advantages and disadvantages of polyclonal and monoclonal antibodies. Okay? So guys, now for the polyclonal antibodies advantages, okay? We have short production time and low cost. Bakit? Kasi malala nyo guys, di ba, dun sa monoclonal antibody production adding, meron tayo nung hybridoma production. So something no that is of high cost. And other than that, we do there in the monoclonal antibody production, di ba, we do isolation. Okay? Dito, kung ano na yung na-produce na antibody, yun na yung gagamitin natin sa pag-produce ng ating reagent antibodies. Okay? So next, or kung ano na yung mga B cells or kung anong klaseng mga antibodies na yung pinroduce ng mga B cells against that antigen, yun na yung gagamitin natin. We do not isolate the B cell to get identical antibodies. Okay, so short production time and low cost, unlike your monoclonal. It's highly stable, okay? High affinity, and of course, it's tolerant of minor changes of antigen. Anong ibig sabihin nito, guys? Guys, di ba malala nyo, sa polyclonal antibody, okay, dito siya, di ba? So, pwedeng ganyan yung antigen, pwedeng ganyan, or pwedeng ganyan. Sa polyclonal, di ba yung mga antibodies natin pwedeng mag-bind sa epitope na square, okay? O kaya sa epitope na bilog ng same antigen natin. Okay? Kasi polyclonal sila. So, iba-iba. They are coming from different types of antigen, of B cell. Okay? So, iba-iba. So, meron din dito. Okay? So, polyclonal sila. Okay? So, ibig sabihin, guys, example, this antigen, okay, will undergo changes. Okay? Yung epitope niya, biglang ang natira guys yan. Biglang ang natira is yung green epitopes lang niya. Yung kunwari, yung round at saka square, nag-iba sila ng itsura. Naging kunwari, naging heart yung round. Illustration lang to guys ha. Okay, sa immunology mas maintindihan nyo siya later. Okay, ito simple illustration or diagrammatic illustration for you to appreciate, okay, or understand yung ibig kong sabihin. Okay, hold on. So ayan, 'di ba dun sa polyclonal, sabi natin we have this antigen here, we have this antigen here, we have this antigen. Tama? Okay? So, ito yung mga antibodies, polyclonal antibodies. Kahit saan sila dun sa antigen na yon magbabind, kahit saang epitope. Mapabilog man yan, rectangle or square, nagbabind sila. They are polyclonal kasi galing sila sa non-identical B cells. Okay? Guys, no, sabi niya, they are tolerant of minor changes to antigen. Okay? Bakit? Kasi kaya nilang i-bind kahit anong klaseng epitope. Example, ito yung antigen natin. Nag-undergo ng change. Yung bilog dati, guys, na epitope, naging heart shape siya. Okay? So, andyan pa rin yung triangle, andyan pa rin yung square. Ganun din dito. Okay? Kunwari may bilog dyan. So, naging heart shape siya or nag-iba siya ng anyo. So, dahil nag-iba siya ng anyo, guys, hindi na siya madedetect nung antibody na against sa bilog. Kasi hindi na siya bilog, hindi na siya yung dati. Nag-iba na siya. Okay? Charot. So, yan. So, guys, it's tolerant of minor change. Bakit? Kasi despite hindi na mabind nitong antibody na to, si bilog, dahil iba na yung itsura niya, nag-iba na siya ng anyo, madedetect pa rin natin if this is the same antigen. Bakit? Kasi polyclonal antibody yung gamit natin. It's directed dun sa iba't ibang epitopes na nasa ating antigen. Okay? Bakit madedetect mo pa rin? Kasi, tandaan mo, hindi lang naman niya ito yung epitope mo. Meron ka pa nung triangle at square. Hindi sila nagbago. So, ibig sabihin, yung polyclonal antibody reagent mo, meron pa siya nung antibody against the triangle and epitope and the square epitope. So, nadadetect mo pa rin siya. So, it's tolerant of minor changes. Okay? Again, okay, that's it. And then for the advantage, it's less sensitive to antigen changes. Ganun yun. Parang dito din, sa explanation ko dito. So kung nag-change yung iyong epitope, okay lang. Kasi polyclonal antibody yan. Ibig sabihin, it's not detecting only a single epitope, but various types of epitope in that antigen. So meron kang antibody na against dyan sa mga epitopes na yan. Okay? And then for polyclonal antibody disadvantage, we have prone to batch variability. What if, for example, yung type lang ng mga antibody karamihan na na-produce dyan sa reagent mo ay yung antibody directed against this one. Okay? So, pwedeng hindi masyadong kita yung result. Diba? Paano kung puro square lang siya? Antibody against the square epitope. Konti lang yung square epitope. So, pwedeng um, tawag dito, less sensitive siya. Ibig sabihin, medyo hindi ganun kita yung result niya. Okay? So, prone to batch variability. Pwedeng mas madami yung antibody sa ganitong epitope or mas konti yung antibody sa ganitong epitope. Okay? Multiple epitopes make it important to check immunogen sequence for any cross-reactivity. Pagdating nyo guys, no, sa immunology and serology ninyo, one of the problems, okay, we have with our testing, okay, or tests, okay, in immunology, serology, is yung tinatawag nating cross-reactivity. Pwedeng magkapareho sila, okay, nung epitope. Okay? So, example, ito for example, yung strain ng isang bacteria o kaya isang microorganism. Okay? And then this is another strain. Okay? Or another microorganism. Okay? So guys, no, meron siyang mga epitope na ganito. Itong bacteria na to, for example. Yan, meron siya niyang mga epitope na ganyan. Ito, nagkataon, ibang microorganism, pero meron siyang kamukhang epitope nito. So tendency, baka iba yung sakit niya, nag-positive siya, kasi pala, meron siyang kamukhang epitope nung isang microorganism. So, dun, yun yung reason guys, kaya meron tayo ng mga tinatawag na false positive result. That's why we usually use this test lang as screening tests. We do perform confirmatory tests to really detect if that's really, okay, the organism causing the infection, yung target organism natin. Okay? So, ganun yung cross-reactivity. You will know more of this in immunology and serology. So, meron siya. It's not exactly the epitope, pero kamukha niya siya. Parang nare-recognize niya na parang siya yun, pero hindi siya, pero magkamukha sila. Nagigets niyo ba ako? So, para siguro pagka nakakita ka ng classmate mo na kambal, first day ng class ninyo, akala mo siya yun, pero yung isa pala yun. Parang ganun. Okay? That's causing cross-reactivity. Okay? Kaya meron tayong mga tinatawag na sa mga screening test na false positive result. Okay? Nagkamali siya ng tanda. Okay? Kasi magkahawig. Okay? Yon. So, polyclonal antibodies, advantage and disadvantages. Okay? So, next, we have this one. The next one is, saan ba yun? Okay, this one. Okay? Next, we have monoclonal antibodies, advantages and disadvantages. So, guys, no, advantages, it's highly specific for only one epitope. Tama? Kasi galing lang sila sa isang population ng B cell. Okay? Immortal hybridoma cell lines have the ability to produce unlimited quantities of antibodies. Bakit? Kasi it's a fusion of your B cell antibody producing cell or your B cell and that of your tumor cell or myeloma cell. Okay? Eh di ba si tumor cell, it has the ability to multiply. Okay? And multiply. So yan, yung kanyang longevity and cross-reactivity ay na-adapt ni hybridoma cell, yung kanyang product. Okay? Or yung hybridoma cell na product ng fusion ni tumor cell at ni antibody producing cell. Okay? And then, minimal cross-reactivity. Bakit minimal cross-reactivity? Kasi isang single epitope lang yung binabind niya. So, if for example, we have here for example, certain strain of E. coli. So, dalawa sila. So, this one has this epitope. Pareho sila meron nung square. Meron din bilog ito. For example, may bilog din ito. Okay? So, kung gumamit tayo, guys, ng polyclonal antibody, ibabind nila yan, ibabind din yan. For example lang ha, kunwari, strain number 1. This is strain number 2. So, kahit if you're using a polyclonal antibody, there's a tendency na meron tayong tinatawag na cross-reactivity. Bakit? Kasi sa polyclonal, yung mga antibodies mo, they are what? Coming from different antibodies, B cell, they are heterogeneous. So they bind different epitopes in an antigen. So ibabind niya lahat. So positive yung test mo for strain 1, magpa-positive din siya with your strain 2. Okay? Kasi polyclonal eh. They have this one, identical epitopes. Unlike when you're using monoclonal antibody. Example. Tignan nyo to guys. So yun. Yung sinabi ko, di ba? Pareho sila. Sorry. Ayan. So, strain 1, strain 2, guys, ha. So, pareho silang mayroong epitope na square, epitope na round. Pero, si strain 1 lang ang may epitope na triangle. So, we're going now, if, for example, the test is, the principle of the test is to detect now, for example, infection caused by strain 1, then we should use, okay, reagent antibodies that are monoclonal. Okay? And these antibodies should be directed to your epitope triangle. Bakit? To make it more specific. Why? Kasi si epitope triangle, nandun lang siya kay strain 1. Wala siya sa ibang strains or dito sa example natin, kay strain 2. Okay? So if you're using a monoclonal antibody, it has, okay, a homogeneous type of antibody. It's directed dun lang sa isang epitope natin. Dito sa picture natin, it's your epitope triangle. So, magpa-positive si strain 1 kasi siya yung dinedetect natin. Walang cross-reactivity. Bakit? Kasi even if you use this sa specimen ng pasyente na may infection with strain 2, negative pa rin siya. Bakit? Kasi yung monoclonal antibody natin, it's directed lang sa triangle epitope. Eh wala nun si strain 2. Okay? So we minimize cross-reactivity and the test is what? More specific. Okay? It's more specific. Okay. Disadvantage, developing a monoclonal takes time and requires high technical skills. Bakit? Kasi nga we have there the isolation, 'di ba, of your B cells. Okay? And then we have also there the fusion of your hybridoma, the fusion of your antibody producing cell and that of tumor cell to produce the hybridoma. So it's costly, it's laborious. Okay, they can produce large amount of specific antibodies, but may too specific to detect in a cross range of species. So, for example, okay, I'll look into the presence of an infection caused by this microorganism. Dahil masyadong specific siya, certain strain lang yung nade-detect niya. All other strains, hindi niya nade-detect, for example, kasi itong strain lang na to ang merong epitope na ganito. Eh, yung monoclonal antibody mo, siya ay directed lang sa epitope na to. Eh, yung iba na strain mo, wala nung epitope na yun. So, hindi mo sila made-detect. Okay? So, ito yung disadvantage niya. Okay? And of course, vulnerable to the change of epitope. So again, example, ito yung antigen natin. O, di ba? So may triangle, may bilog, may square. Okay? So yung monoclonal antibody natin para sa sakit na to ay directed against your triangle. Okay? Antigen. What if nagkaroon ng change sa epitope due to factors like for example nagkaroon ng mga ah, ah, tawag dito ah, evolution, okay, nung microorganism due to, for example, environmental exposures, resistance, mga ganyan. Okay? So, kunwari, naging ganito na siya. Meron pa rin siya nung bilog na epitope, meron pa rin siya nung square na epitope, pero yung triangle niya, for example, naging ano ba? Naging hexagon, for example. Yan. Sorry sa drawing. Hexagon ba yan? Wait lang Kunwari, naging, ano na lang siya, ano ba? Naging, ano bang shape? Kunwari, naging heart na lang. O, di ba? So, the monoclonal antibody that we are using, okay, is the same. It's directed against the triangle. Pero dahil nagkaroon ng change dun sa epitope natin, magkakaroon ka ngayon ng false negative result. Bakit false negative? Hindi niya na ma-detect, hindi na makakabind itong antibody na to kasi siya ay directed against the triangle epitope. Pero dahil nagkaroon ng change yung ating antigen or due to, for example, evolution of the microorganisms or exposure to substances or environment that causes, for example, changes, for example, mga ganyan, or antibiotic resistance. So factors, okay, that may lead to the, how do you call this one, the change of the epitope. Okay? So ito, dahil directed sana siya sa triangle epitope natin, 'di ba? Eh nagkaroon ng change yung itsura niya. So hindi niya na ma-recognize. Okay? So walang binding na mangyari. So that leads to false negative. Okay? So yan yung disadvantage naman ni monoclonal. So if you can see guys, no, yung disadvantage ni monoclonal are actually the advantages of polyclonal. And the advantage of monoclonal are actually the disadvantages naman of polyclonal. Okay? Antibodies. So that's your monoclonal versus your polyclonal. So guys, no, if you have questions, feel free to raise them in the forum discussion. Okay, I'll be answering them. Okay. So again, this is your direct and your indirect immunofluorescence. Okay, so let's just have a review. Guys, tandaan nyo, sa direct immunofluorescence, we only have single antibody and we call this your primary antibody. Your primary antibody is directed against your antigen of interest. Your primary antibody has the label. Okay, in your indirect immunofluorescence, we have two types of antibody. We have the primary antibody. Again, the primary antibody is the one directed against the antigen of interest. Other than the primary antibody, we have the secondary antibody. It is the secondary antibody that is labeled, okay, in your indirect. Okay, unlike in your direct, it is the primary antibody that is labeled. Okay, again, you in the indirect immunofluorescence, your primary antibody is directed or is against your antigen. Okay, And your secondary antibody naman is the one against naman sa iyong primary antibody. Okay? So, if you can see here, nagbabind siya. Yan. Okay? So, anong advantage ni direct ni indirect versus direct? Of course, si indirect, it's highly or is more sensitive than that of your direct. Kasi amplified yung result. So imagine this single label compared to this one, okay? Na pwedeng dalawang signal yung makita mo indicating a positive result. Okay? So that's your direct immunofluorescence siya kasi nga ang ginamit nating label, guys, ay fluorochrome. Okay? And we're viewing the result under your fluorescence microscope. Okay? If the label is, for example, how you call this one? For example, an enzyme. Okay? So we have an enzyme immunoassay. Okay? So we can read the result using colorimetric methods. Pwedeng color change. Okay? So that's your um, direct and indirect immunohistochemistry. Okay? So next. Again, different types of labels that we can use. We have radio labels, radioactive labels. Pwede yon. O kaya naman fluorochromes. O kaya naman enzymes. Okay? O kaya naman mga latex particles. Okay? So you will know more of that pagdating nyo, guys, sa immunology and serology ninyo. Okay? Okay. So yan. So we have here the applications of your immunohistochemistry. We have prognostic markers in cancer, tumor of uncertain histogenesis, metastasis, okay, response to treatment, infections, neurodegenerative diseases, muscle diseases, and brain trauma. So you will know more of this pagdating nyo sa immunosero, lalong-lalo na guys, no, dito sa infections natin. Okay? For cancers sa histopath ninyo. Okay? So, yeah. Next. Okay, so guys, we have here an application. Kamukha lang ito guys kanina nung fluorescence assay ninyo. Ito din siya, oh. It's using then fluorochrome. So it's fits labeled. Kung mapapansin nyo, we have this apple green color here. Okay, so this is an immunocytochemical method. Immunohistocyto naman to because we are actually using the cell. Okay, so cell yung application natin. Okay, so if you can see in letter A, okay, this is a decidual cell grown in vitro. And then we have stained it, okay, to reveal a mesh of intermediate filaments throughout the cytoplasm. So guys, no, the primary antibodies, okay, that were used here were actually directed against your desmin. So si desmin ng intermediate filaments, guys, no, ang dinedetect natin dito. Okay, siya yung antigen natin. And this letter A technique here is actually indirect. Why? If you can see here, it is the secondary antibody that is labeled with your fits. Okay, so your secondary antibody now is directed against your primary antibody. Okay, while yung primary antibody naman ninyo guys, siya ay against doon sa desmin ninyo. Okay, so that's your Okay. So guys, again here, in this letter A technique, it's an indirect technique. So yung primary antibody, it's against the desmin of the intermediate filament. And then we can detect the binding of the primary antibody with the desmin through the use of your secondary antibody. So it's the secondary antibody that is labeled with your fits. So yung nade-detect na ngayon natin is their binding. So ayan siya. Okay. So yan, na-detect na yung ating intermediate filaments through our protein desmin. Okay? So kung makikita nyo guys, yung dinedetect specifically is yung desmin natin. Okay? So in the next, we have here the letter C picture. Okay? It's a section of your pancreatic acinar cells. And this is a view under your transmission electron microscope. Okay? Incubated with antibody against the enzyme amylase. Okay? And then with a protein coupled with gold particles. Okay? So guys, no, ayan. We are actually detecting naman here yung mga dots, kung makikita nyo. Okay? So that's the application of your immunohistochemistry okay, in your tissues, or we call it immunocytochemistry when we're referring to its application sa cell. Same principle, okay? So next, we have here the hybridization technique. We have here, okay guys, the hybridization techniques, okay? So guys, no, yung hybridization technique, same lang siya with your immunohistochemistry. We use this, okay, to visualize molecules in the tissue, okay? So it allows the specific identification of sequences, okay, of DNA or mRNA, okay? Okay, so ito naman guys, no, is detection of your DNA or mRNA sequence. Okay? So this is through the use naman of complementary strand of a nucleotide probe. So di ba kanina guys, malala niyo yung label or the antibody, di ba? We use the antibody to detect the antigen. Dito, we are using, okay, or we detect, I should say, okay? Dun kanina sa immunohistochemistry, di ba, we're using, how you call this one? The antibody to detect this antigen in the tissue or in the cell, okay? In this technique naman, we are using a complementary strand of nucleotide. We call this your probe in detecting now the unknown, okay, nucleotide sequence. Okay? So, yeah. It's commonly performed with nucleic acids in solution. Okay? So guys, no, an example of technique is your in situ hybridization technique. Okay? So solution of nucleic acid are applied directly to cells and tissue sections. Okay? So let's look into this one. Okay? So we have the following nucleotide probes. Again, yung nucleotide probes, guys, these are complementary nucleotide sequence that we will be using to detect, okay, the unknown nucleotide sequence. Okay, so example, we have the oligonucleotide probes, single or double-stranded DNA probes, and RNA probes. Okay, so labels that we use, again, di ba kanina dun sa immunohistochemistry nyo, yung mga antibody natin, na meron silang labels. Ganun din dito. Yung mga nucleotide probes natin, guys, may labels sila. They can be your radioactive isotopes. Kapag radioactive isotopes yung ating probe, binabasa natin yung result using your autoradiography. Okay? Kapag ka naman yung digoxygenin, biotin, okay, mamaya makikita nyo yung mga techniques, okay, in reading naman their results. Okay, so tignan natin na. Yan. Yan. Okay, so guys, no, you will have a discussion more of this pagdating nyo sa microbiology, but this one is a simple, okay, overview of, or an overview, okay, of this procedure. So these are the basic steps in hybridization assay. Okay, so let's look into the procedure. Number one is the production and labeling of the single strand nucleic acid probe. Okay, and then after that is the preparation of the single strand target nucleic acid. Okay? So ito yung labeling of single strand nucleic acid probe. Ito yung gagamitin natin guys to detect itong target nucleic acid natin. Okay? So again, when we say labeling, we can either use your fluorochromes, your radioisotopes, yung mga nandito kanina, your biotin, avidin, okay? Streptavidin, okay? So you will know more of that. Pagdating nyo guys, no, sa micro nyo, meron yung discussion doon, okay? And then mixture and hybridization of target and probe nucleic acid. Then after that, we're going to combine them, okay? So dito sa histology, we simply add the the nucleic acid probe doon sa tissue. So if it is, okay, if for example yung target nuclei natin ay siya talaga yung hinahanap natin, yung unknown nuclei natin ay nucleic acid natin ay siya yung target nucleic acid natin, magkakaroon sila ng binding. And then finally, if there's binding, we will detect the hybridization. If we have used radio label or radioactive labels, then we should use autoradiography in reading the result. If we have used fluorochrome, then we use fluorescence microscopy in reading the result. Okay? So, ito yung example niya, guys. Para mas maintindihan nyo, we have the diagram here or yeah, we have the picture here. So, ito siya, guys. Okay? So, guys, no, hold on. Wait lang nga, guys. Kasi meron yung, ano ba to? Hindi ko makita dahil meron yung, ano, for annotation na mga, ano. Okay? So, yan. So, guys, no, we have here, okay, pakinggan niyo ako. So, we have here, no, the probe. Ito yung probe, ito yung gagamitin natin to detect the unknown. Okay? Example, this is the unknown 1 and this is the unknown 2. So, to have a positive result, your probe should be complementary to your unknown. If it's not, then that's negative result. Okay? So example, nagkaroon ng food poisoning. Okay? And then we want to detect what specific strain of E. coli is causing the infection. For example, E. coli siya, but we have to determine kung anong strain. Okay? For us to know ano ba yung appropriate treatment na dapat nating ibigay dun sa patients natin na na-food poison. Okay? So guys, no, example, so we're going to create a probe against complementary to the nucleic acid sequence of a certain E. coli strain. Okay? So guys, no, ito siya, yung kanyang sequence, AAAGGGG. Alam niyo to, 'di ba, sa biology niyo noon. Okay, yung um uracil, 'di ba? Sequence natin, mga ganyan. Okay? Guanine, thymine, ganyan. Okay? So, after that, okay, this probe will be combined doon sa tissue. For example, ito yung first specimen natin. This one is our first specimen. Okay? If they're complementary, guys, magbabind sila. Complementary ba siya? A is to TA. O yan. They're complementary. So, ngayon, what will happen? They will form a duplex. Magbabind si probe mo dun sa unknown. Okay? Ito yung unknown. Okay? So, this is the probe that we use to detect, okay, the unknown target. This is the target or the unknown, I should say. Okay? So, if the unknown, okay, is complementary to the target, sorry, if the unknown is complementary to the probe, they will bind to form the duplex. Okay? So, the combination or the binding of your probe to that of your target is what we call your duplex. Okay? Then, finally, okay, we read the result. If gumamit tayo, guys, no, ng fluorochrome, then we use the results of fluorescence microscope. Okay? And eventually, kunwari, ito si probe for E. coli strain 0157. Okay? At nag-form sila ng duplex. Okay? Ibig sabihin, pag niread mo siya sa fluorescence microscope, after washing, matitira, okay, yung probe mo kasi nakafix na siya with your target nucleic acid sequence. Okay? So, hindi kahit mag-wash ka, hindi matatanggal kasi nakafix na siya dun sa nucleic acid sequence sa specimen mo. Okay? So, pag dinetect mo siya sa fluorescent microscope, makikita mo. Kung fluorochrome siya, for example, using your feeds, makikita mo yung apple green doon. Ibig sabihin, ang organism na nag-cause ng infection is your E. coli 0157. Halimbawa lang nga, okay? So, kunwari guys ha, yung probe kasi na ginamit mo ay para dun sa strain na yon. Okay? O for example naman, yung another specimen mo naman. Okay? Ito naman. So, guys, no, ito yung probe mo to detect if it's E. coli 0157. At complementary ba sila? Hindi. Okay? So, kung makita mo A, ito C. It should be T, di ba? T, T, T. Complementary to this is C. Yan, iba-iba. Okay? So, hindi sila magbabind. So, what will happen? Dahil hindi sila complementary or they are non-homologous, okay? Your target and your probe will not form your duplex. So, there is no duplex formation. Pag nag-wash ka ngayon, guys, matatanggal yung probe mo kasi hindi siya nakafix doon sa sequence or nucleic acid na nandoon sa iyong tissue or cell. Okay? Kasi hindi naman pala present or hindi naman pala si 0157. Okay? yung organism, for example, na yon. Okay? Or yung target mo, okay, na hinahanap na nucleic acid sequence. Okay? So if you view the result dun sa microscope, wala kang makikita. Negative. Okay? So that's for your hybridization. Okay? So ulitin ko guys ha, for to to have a formation of your duplex, your probe nucleotide sequence should be complementary to your target. If it's not complementary, there will be no duplex formation. It means negative. Okay? Or the unknown organism is not identified. Okay? So you can use another probe. Baka ibang strain siya, okay, ng microorganism. Okay? So, ganun siya until you find the exact probe that will tell you kung anong exactly siya na strain or microorganism. Okay? So, guys, no, this one, you need not to memorize this, but this just only shows no yung mga steps na ginagawa natin. Okay? So, we have here, guys, no, if you're using a radioactive label, okay, we use the result using autoradiography. So, we use it or we are reading the result just like an X-ray film. So, autoradiography, parang ganun siya. Okay? In your, malala nyo yung autoradiography kanina, yung positive result, di ba, is indicated by, how do you call this one, black areas, okay, doon sa iyong specimen. Or since you are using X-ray, doon sa film makikita mo, okay? And then, if you are using biotin avidin as a label for your probe, ito sila, okay? Then, you use colorimetric detection. Basahin mo yung result using colorimetric assay, okay? So there is what? Color change. Okay? So next, if you are using naman acridine labeled probes, okay? So acridinium yung label mo, okay? So ibig sabihin these are your chemiluminescent reporters. Then you are going to read, okay, the result using chemiluminescent assays. So you are going to read the result, okay? Or the result, I should say, is indicated, okay, by the capturing emission of light. Okay, by the detected capturing emission of light. Okay, so ulitin ko, Ading, if the label used is, or the label used is a radioactive reporter, yung label, we call it a reporter molecule. Okay, these are your reporter molecules, yung label ng probes natin. If it's a radioactive reporter, then we're using autoradiography, the use of your films. If you were using biotin avidin reporter, we can use colorimetric detection, okay, to detect now the result. Okay, if we're using acridinium or chemiluminescent reporters, then we should use now detection of emitted light or yung chemiluminescence. Okay, so that's for your hybridization. Guys, no, example of application we have here. Nakita nyo to? Okay? So, dito, this is an example of your fluorescence in situ hybridization technique, okay, in prenatal screening. Okay? So, guys, no, kung makikita ninyo, wait lang, yung, how you call this one? Itong mga green na bilog na ito, these are your fluorochromes, okay? These are actually detected using now yung iyong reporter molecule, fluorescent reporter molecule na ano, fits. Okay, your fluorescence thiocyanate. This one naman are your um, stained by your acridine orange. Okay? So, itong mga to yung mga orange dyan. Okay? So, guys, no, if you can see, the orange probe, okay, na ginamit is labeled, okay, nakita mo yon. Wait lang ha, guys. In-erase ko yung... Okay. So, we have there, di ba, may nakikita kayong kulay na dalawa, orange at saka green. Ading, anong ginamit dito to detect yung chromosome na may problema? Okay? Gumamit sila, guys, no, ng nucleic acid probe, okay, against your chromosome 21 and chromosome 13. Okay? Guys, yung green, yun yung chromosome 13 ninyo. Okay? Bakit naging green? Kasi yung complementary probe na ginamit ay merong reporter molecule na fluorescent or yung tinatawag mong fluorochrome na fluorescein isothiocyanate. Kaya apple green yung kulay niya. Okay? So this is your chromosome 13 detected. Okay? Yung orange naman, guys, okay, ay yung kulay ng inyong rhodamine. Okay? So, ang nangyari, dinedetect naman niya ngayon, okay, yung probe natin na orange, dinedetect niya ngayon yung ating nucleic acid sequence para sa chromosome 21. Okay? So, ibig sabihin to mga orange na to ay chromosome 21. Okay? So, guys, no, ano to? Okay? Guys, no, these are your fetal cells. These are actually derived, okay, dun sa amniotic fluid ng mga nanay. Okay? Na sa yeah, nabuntis, okay? So yung amniotic fluid nila, it contains fetal cells. Of course, the slow cells coming from the fetus. This one here, dito sa my right side, normal. Bakit? Kasi kung makikita nyo, di ba, we have two copies of the chromosomes, okay? So we can see here two green. Ibig sabihin, meron siyang um, two signals for your chromosome 13, while for your chromosome 21, meron din siyang dalawa. So, normal. Okay? But here, dito sa left side, this is a fetal cell of a baby that actually, or a fetus, okay, that actually have trisomy 21. Okay? So, this fetus, pag pinanganak siya, alam na natin meron siyang trisomy 21. So if you can see, yung chromosome 13 niya, dalawa. Yeah, that's normal. Pero pag pumunta tayo dun sa orange signal, tatlo. Ibig sabihin, there are three copies of your chromosome 21. And that's what we call, guys, no, your trisomy 21. Ibig sabihin, yung fetus na to, okay, or the fetus that owns this fetal cell, okay, dito sa first specimen natin, ay magkakaroon ng Down syndrome or merong Down syndrome. Okay, makikita natin yun pag napanganak siya. Okay, dito normal kasi meron lang siyang two copies of each chromosome. Okay, so yun ang example ng application ng ating in situ hybridization. So, anong advantage nito? Ibig sabihin, hindi pa pinapanganak si baby, na-detect na natin na ito yung possible na magiging sakit niya. So, we can give early management, okay, to at least reduce, okay, the harm, or at least know how to manage, okay, our future baby or the patient, okay, to avoid now further damage, okay, sa kanya. Okay? So, that's the application of our in situ hybridization. So, na-detect natin. Okay? For microorganisms, yun yung example ko naman sa inyo, detection. For example, na-detect mo anong exactong microorganism yung causing infection, or more specifically, anong strain. Okay? So, that's your hybridization technique. Okay? And then last, we have interpretation of structures in tissue sections. So guys, no, ito, work po ito nung ating mga pathologies. Okay, work po ito nung ating mga pathologies. So, like for example, yung mga diniscuss natin kanina guys na procedure, di ba, in tissue preparation. So kung nagkamali ka, sa procedure mo, may mga pwedeng andon, pwedeng result dun sa tissue mo. Like for example, madumi yung stain mo. So may mga precipitates na pwedeng ma-add. So nare-recognize, okay, nung pathologist, alin yung artifacts lang. Alin yung mga hindi hindi results ng sakit talaga or nagkaroon kasi may mali sa procedure. So, hindi natin to work ito ng mga histologists or pathologists. So, they can determine no, if these molecule, for example, seen under the microscope, artifact lang o kaya precipitates lang ng stain natin. Okay? O kaya naman ay at tawag dito, merong clinical significance. Baka it means something. Okay? Or it may indicate a disease or cancer. Okay? So something like that. So interpretation of structures sa ating mga medical technologies, we do not, okay, do read no, yung mga slides natin. Okay? Ang gumagawa niyan lalo na in cancer diagnosis ay yung mga pathologists natin. Okay? So they usually do the reading. Okay? So those are the different methods of histology. Okay? So guys, no, for the yeah, for the interpretation of structures in tissue sections, Okay, you may watch it, sorry, here in sorry, in the, where is that now? Yeah, in the immunohistochemistry, okay, we have the link there where you can watch, okay? Kanina kung nakita nyo yung link, okay, pwede nyong makita yung protocol on how direct and indirect immunofluorescence are performed in the laboratory, and even manual immunohistochemistry in the laboratory. How is it performed? Okay, how it is performed in the lab. Okay, so please to watch that. Okay, so guys, now let's have a recap of the things that we have discussed. Okay, so we have discussed about the overview of methods used in histology. We have discussed about microscopy. Okay, we have two types. We have the light microscope and the electron microscope. So under your light, we have the different types like your bright field, your dark field, your confocal, your phase contrast, okay? And the other counterpart, we have the differential interference, okay? And for your electron microscope, we have two. We have your SEM and your TEM, your scanning electrons, scanning the surface. Your transmission, we're able to examine, okay, the inner, okay, structures of the cell of or of a microorganism, okay? So next, and then, yeah, next we have preparation of tissue. So we have discussed about these 10 steps. Fixation to preserve the tissue. Most commonly used, we have the 10% neutral buffered formalin. We also have dehydration from the term itself to remove the cellular, intercellular, or extracellular water, okay, from the tissue. So we're using here increasing grade of alcohol. Most commonly used, ethanol, okay? And then we also have clearing. Okay, so clearing is also known as your dealcoholization. The purpose of that is to remove what? The alcohol from the tissue. Okay, most commonly used, styrene. Okay, infiltration, that's the impregnation of tissue with your paraffin wax. So most commonly used medium is your paraffin wax. Okay, and then the embedding is also known as your casting, where we produce our tissue block by placing the previously infiltrated tissue in the mold, right? And then we create, okay, a block by pouring, okay, molten paraffin wax doon sa ating mold, okay? And then creating now a tissue block. And then we have trimming and then section cutting to produce now thin tissue ribbons, okay? And finally, we have staining. In the staining method or routine techniques, we usually use your routine H&E or hematoxylin eosin staining technique. Okay, so in the H&E technique, your nucleus is usually dark blue or blue-black, and then your cytoplasm is pale pink in color. And then for mounting, okay, that's to make the slide, okay, for permanent keeping. And of course, to avoid fading of the stains, we do mount the slides using now a mounting medium. The commonly used is your Canada balsam, which is an example of a resinous media. And then finally, labeling, okay, our slide. Okay, so next, okay, we have also discussed autoradiography. So it talks about localization of your macromolecules. Where in the cell are these macromolecules, okay, being synthesized? Okay, so next, cell and tissue culture, like for example, it's used in microbiology to culture now your virus, okay, in cells, or for example, in studies of your tumor or cancer cells, okay, to develop drugs, okay, for the treatment of certain cancers, okay, types of cancers. Enzyme digestion, okay, the use of this is of course to confirm identity of the stain material, if it's really glycogen, DNA, or RNA. So we have there the use of your DNase or deoxyribonuclease in enzyme extraction for DNA, and your ribonuclease for enzyme extraction of your RNA. So please memorize din the color result with your Fuelgen. For Fuelgen, DNA is always red. Okay? But when you extract it, it becomes negative kasi natanggal mo si DNA. Okay? Metal green pyronine, your metal green is stains now your DNA green, and your pyronine stains your RNA red. Okay, but when you've performed RNase extraction, ang ma-stain mo na lang ay DNA ng green color. Your RNA will be negative. Okay? So next, we also have discussed about enzyme histochemistry. So dito, we're talking about the localization of enzyme activity. Where exactly in the cell or in the tissue, okay, this enzyme, okay, doing its activity. Kamukha guys nung ano ulit? Autoradiography. Okay? Diba sa autoradiography, we're talking about the synthesis of macromolecules. Where exactly in the cell or in the tissue these macromolecules are being produced. Okay? So dito naman, we talk about enzyme activity. Okay? So next, we also have discussed about immunohistochemistry and hybridization technique. Guys, no, sa immunohistochemistry, please don't forget the difference between direct immunohistochemistry and indirect immunohistochemistry. Again, in your direct immunohistochemistry, we're using primary antibody only. It's the primary antibody that is labeled, while in the indirect immunohistochemistry, it's the secondary antibody. Okay? In the direct immunohistochemistry, the antigen is detected by using a labeled primary antibody. The primary antibody in your direct immunohistochemistry is directed against the antigen of interest. Okay? In your indirect, okay, the antigen is detected, or the antigen, okay, is actually, or the primary antibody, I should say, will bind the antigen of interest, but this binding will be detected through the use of a secondary antibody. So it's the secondary antibody that has the label. Okay? So your indirect immunohistochemistry, okay, gives now a more sensitive tests, okay, compared to your direct immunohistochem. Okay, kasi amplified yung result natin sa indirect. Okay, and don't forget also the difference between your polyclonal antibodies and monoclonal antibodies, how your polyclonal antibodies are produced, and so with your monoclonal. Your polyclonal, again, they are directed against different epitopes in an antigen. Your monoclonal, they are directed against a single epitope. So they are more specific in detection. Okay? But again, the disadvantage is that it's what? It's prone to change in your epitope. Okay? So yan. So again, the polyclonal antibody production and the monoclonal antibody. Don't forget, okay, that this procedure is, of course, costly. Other than that, it's a bit laborious compared to your polyclonal antibody production. Bakit? Kasi in your monoclonal antibody production, we have to what? Produce first our hybridoma. And your hybridomas are the ones producing now your desired antibody. Hybridomas are combination or fusion of your antibody forming cells and tumor cells. Okay? And then, yeah, we have there the difference and the advantages and disadvantages of monoclonal and polyclonal antibodies. Okay. And then finally, guys, we have the hybridization technique, the use of your probes to detect target, okay, nucleic acid sequence. So we have the nucleic acid sequence probe, okay, that we use. Okay, they are actually labeled using yung tinatawag nating mga reporter molecules. So your reporter molecules, pwedeng radioactive isotopes. We have your chemiluminescent reporters o kaya mga fluorescent, okay, reporters or fluorochromes. Okay? So, yan. We have here the basic steps in hybridization assay. Okay? And then, of course, last is the interpretation of structures in tissue sections. Okay? So, guys, no, those are the different methods that we use in the study of histology. So guys, no, kung hindi nyo na-visit no, yung mga link dito sa videos na to, kindly just visit na lang doon sa ating suggested self-directed activity section. And also, do not forget, okay, to try our microscopy simulation. Why? Kasi sa microscopy simulation, andun yung viewing ng mga different slide samples natin. So it's important for you pala guys ha, na i-view lalo na yung letter E doon. Okay? So please try viewing that in our virtual microscope environment. Okay, so guys, no, that's all for our topic, okay, this week. So see you, or I hope, okay, you will listen to topic 2, okay, for the next week. That's all.